The first speaker is Ms. Priya Hariharan, and the topic is spectrum of erythroid phenotypes generated by the KLF gene 1 variants in patients with hemoglobinopathies. Is she here?
Good afternoon, everyone. I'm going to talk on spectrum of erythroid phenotypes generated by KLF1 gene variants in patients with hemoglobinopathies. So KLF1 gene is located in chromosome number 19, and it consists of 3 exons which codes for 362 amino acid KLF1 protein. The protein contains 2 functional domains, the transactivation domain and the zinc finger domain that binds to the GC-rich DNA motifs. So KLF1 being the transcription factor regulates major aspects of erythropoiesis. So this is the string model showing the interlinked role of KLF1 with associated genes. Thus it plays a major role in erythroid lineage commitment, activates the expression of various erythroid-specific genes. It plays a role in direct globin gene switching, that is switching from fetal to adult hemoglobin synthesis, and it plays a role in terminal erythrocyte maturation. So variations in the KLF1 gene leads to differing phenotypes from several benign hematological conditions like high HbF, altered blood group phenotypes, then borderline HbA2, to a more severe hemolytic anemia condition. So this diagram specifically represents the regulation of KLF1 gene in hemoglobin switching. So under normal condition, the KLF1 protein activates beta globin gene synthesis, and it suppresses the gamma globin gene expression through another transcription factor, that is BCL11A. Under mutant condition, however, the switch process is partially reversed by activating the excess gamma globin gene expression. So with this background, our aim and objective was to screen for the KLF1 variants and to find its association with erythroid phenotypes to elucidate the role of KLF1 along with BCL11A in regulating the gamma globin gene expression. So this was the study group. We screened for the KLF1 variants in 410 individuals, of which 100 were sickle cell anemia patients, 100 were beta thalassemia homozygous patients. Rest are the other patient groups along with 50 normal controls. This was the methodology followed, that is we did primary screening followed with molecular analysis of KLF1 gene, gene expression studies, and in silico analysis to detect the effect of the variants on protein structure and function. So this is the results. This table represents the hematological analysis of the patient groups studied in our study. So coming to this molecular analysis of the KLF1 gene, this shows the schematic representation of exon-wise distribution of KLF1 variants. Overall, we identified 15 variations, of which 6 were found to be novel. The overall incidence of KLF1 was found to be around 8.1% in our study, with codon 102 T2C variation being the most common SNP in our study group. This is the graphical representation of KLF1 variants in our patient group, and we identified that the promoter variation, that is minus 148 G2A, is the most common variant in our population, followed with codon 182 T2C variation and codon 5 G2A variation. Rest of the variants were at a frequency of 0.2%. Then we tried to associate the KLF1 variants with differing phenotypes. So as raised HbF level ameliorates the clinical severity of hemoglobinopathies, the association of KLF1 variants with HbF level was first studied. So in sickle cell anemia patients, 10% of SCA patients showed presence of KLF1 variation, and the patients having KLF1 variant had increased raised HbF level. Similarly, in Swiss type HbFH patients, 13.1% cases showed KLF1 variants, and similarly, the patients with mutant KLF1 had increased HbF level. It was also interesting to find that the KLF1 variants were significantly higher in thalassemia intermedia group, which is the milder patient group in beta thalassemia homozygous. as compared to the thalassemia major, that is the severe group. Then coming to borderline HbA2 phenotype, in the borderline HbA2 group, 7.6% showed KLF1 mutation, and it was interesting to find that in this group, patients with KLF1 variants had slightly elevated HbF level as compared to those having normal KLF1 gene. Similarly, in Swiss type HPFH group, the patients having KLF1 gene mutation showed slight elevation in HbA2 levels as compared to those having normal KLF1 gene. And interestingly, we also came across a patient who had a raised platelet phenotype with a platelet count of 11 lakhs, and he was identified with a novel KLF1 variation which generated a premature stop codon at 358 amino acid position. Then to validate our studies of KLF1 variants having a role on HbF induction, we carried out gene expression studies. And we found that patients having the KLF1 variation showed decrease in the KLF1 gene expression along with BCL11A expression, with especially the hemoglobinopathy patients showing inverse increase in gamma globin gene expression. So this suggests the interlinked role of KLF1 and BCL11A in controlling the gamma globin gene expression. Now to study the in silico effects of KLF1 variants on the protein function and stability, we found that out of 9 coding variants, 5 variants had possibly damaging effect on protein structure and function, and also they showed decreased protein stability except for 1 variation. Also, the 4 promoter variations which we found had a transcription binding site. So this suggests that these variants may alter the transcription factor binding to the promoter region. Then we came across the polar interaction among the different KLF1 variants, and we found that the mutant KLF1 had increased polar interaction with the surrounding amino acids, thus may be decreasing the protein stability. So to conclude and to discuss, the KLF1 variants are linked with wide range of loosely associated phenotypes. In our study, we detected that the KLF1 variations are associated mainly with benign phenotypes like raised HbF levels, borderline HbA2, and these mutations were prevalent in milder cases of hemoglobinopathies, thus enlightening the role of KLF1 in induction of HbF levels and in reducing the disease severity. In this study, we also showed an interlinked role of KLF1 and BCL11A on regulating the HbF levels, and thus it may be considered as a therapeutic target for positively boosting the HbF levels. Also, the patients had a borderline HbA2 phenotype. So, thus KLF1 screening must be considered, especially in borderline HbA2 cases in beta thalassemia screening program. And to conclude, this is the first study in our Indian population which identified KLF1 variants. However, a large-scale study may help in precise association of these variants with differing phenotypes. So with this slide, I would like to say that it's a need of an hour to identify more modulators of HbF level that would ultimately help in clinical management of hemoglobinopathies. Thank you.
Very nice presentation.
Thank
but
you,
you used the terms variants, mutants, all interchangeably. So you need to have clarity on that. Okay. And also you talked about as if all the variations lead to high F. We really don't know that. Many of them could be just polymorphisms. And also the association with the high platelet count, that looks very odd. I don't think you can implicate KLF1 for that. There could be some other pathogenetic mechanism giving rise to that, Any questions? We are short of time. I think you've exceeded your time, but is there any query? So if there are none, I think we'll go to the next talk. Thank you very much.
Thank you, ma'am.
So we have Dr. Ekta, who's going to talk on the genotype-phenotype correlation of deletions associated with delta beta thalassemia, hereditary persistence of fetal hemoglobin, and hemoglobin lepor.
Good afternoon, everyone. The topic for presentation today is genotype-phenotype correlation of large deletions in the beta-globin gene cluster. Large deletions in the beta-globin cluster involve delta and beta-globin genes with or without the deletion in the gamma-globin genes, and they usually have increased HbF levels. More than 50 different large deletions are known with varying 5' and 3' breakpoints in the beta-globin gene cluster, with significant phenotypic diversity observed among them. The most common types in the Indian population are HPFH3, delta beta not thalassemia, which is the Asian Indian deletion inversion type, and the hemoglobin lepor. Molecular characterization of these deletions is important for accurate diagnosis and management. The aims of my study is to evaluate the spectrum of large deletions in beta-globin gene cluster in the Indian population and to study the hemogram and HPLC parameters along with genotype-phenotype correlation in such patients. This is a retrospective study including the molecularly confirmed cases of large deletions in the beta-globin gene cluster from January 2002 to July 2018, presenting to our department in the CMC Medical College, Vellore. Individuals with different HbF or HbA2 on HPLC were taken. The ones which when the criteria was that the F level and A2 levels were such that it was not fitting into the category for beta thalassemia. So we suspected a deletion in such patients, and quantitative fluorescence multiplex PCR, which is also known as the gene dosage, was done on such patients. If the gene dosage showed deletion, if it was positive for deletion, then appropriate gap PCR available in the department was done, which was for hemoglobin lepor, HPFH3, and delta beta not thalassemia. If gap PCRs were negative, we suspected some rare deletions, and so beta MLPA was done to detect the rare deletions. To summarize the results, we observed 235 individuals with large deletions in the beta-globin gene cluster, out of which 65 were parents, and all the parents were heterozygous for these deletions, mainly for heterozygous delta beta, heterozygous HPFH3, heterozygous lepor, and heterozygous for rare deletions. 170 patients were diagnosed, and out of which 57% was delta beta thalassemia, 21% was HPFH3, 17% was hemoglobin lepor, and 5% were rare deletions. Now, to break up among the each group, among the delta beta group, 19% was heterozygous delta beta, 23% was beta thal delta beta, and 15% was homozygous delta beta. Among the HPFH3 group, 12% was heterozygous HPFH3, and 9% was compound heterozygous for beta and HPFH3. Among the hemoglobin lepor group, 5% was heterozygous lepor, 8% was beta thal lepor, and 4% was homozygous lepor. On comparing the lab parameters among the three heterozygous conditions, we observed that all the three had normal hemoglobin values. However, heterozygous delta beta and heterozygous hemoglobin lepor showed microcytosis as well as hypochromia, while heterozygous HPFH3 had normal MCV and MCH. This is a bubble graph showing the mean HbF and A2 values of the two conditions, that is heterozygous HPFH3 and heterozygous delta beta. And we observe significant difference in A2 and F between the heterozygous HPFH3 and heterozygous delta beta, with a mean F of 17.8 in heterozygous delta beta and a 30% of F in heterozygous HPFH3. On comparing the lab and the clinical parameters among the homozygous, that is homozygous delta beta and homozygous lepor, we observed that both presented with similar degree of anemia, and we observed no homozygous HPFH3 in our study. On comparing the lab parameters in the compound heterozygous conditions, that is beta delta beta and beta HPFH3, we observed that beta delta beta had an earlier age of presentation with a lower mean hemoglobin percentage and a higher ferritin level compared to compound heterozygous for beta and HPFH3. The same bubble graph again shows the HbF and A2 mean values of beta delta beta and beta HPFH3, and we can see that both these conditions cannot be differentiated based on A2 and F levels, and this may underscore the need for molecular approach for an accurate diagnosis, prenatal diagnosis, and genetic counseling. On comparing the delta beta group, that is homozygous delta beta and compound heterozygous for beta delta beta, we observed that beta delta beta had an earlier age of presentation and higher proportion of patients presented with hepatosplenomegaly compared to homozygous delta beta. One point to note is that both these conditions presented with features of hemolysis, and the blood picture showed extreme anisopoikilocytosis and microcytosis, which can be confused with other causes of hemolytic anemia. And so HPLC and mutation analysis may help in differentiating them. As I have already mentioned, we had eight cases of rare deletions in our study, and these are the HbF and A2 values in those conditions. And as we can see, the F values were raised in the which falls in the category of homozygous beta thalassemia, and so such patients can be misdiagnosed as homozygous beta thalassemia on HPLC. But when we compared the HbA2 and F values of their parents, we saw that the A2 and F values were around 6 to 9% in the parents, and which are not consistent with the diagnosis of beta thalassemia trait. This warrants the need for molecular diagnosis, emphasizing the importance of parental screening in patients with suspected beta thal major. The right side picture shows the 5' and the 3' breakpoints of the different deletions in the beta globin cluster. To emphasize further the role of molecular analysis in rare deletions, we took I want to present an example, 2-year female transfusion dependent and hepatosplenomegaly. The patient had an F of 93.5%. The parents had an F and A2 values of 6 to 7%. So we suspected a deletion in this patient. So a gamma delta beta gene dosage was done on the patient and the parent's DNA. The patient showed absence of beta peak, while the parent showed heterozygous beta deletion. To further characterize the type of deletion, beta MLPA was done, and the patient showed a homozygous beta deletion, 12.6 kb deletion Dutch 1 type, while both the parents showed heterozygous beta deletion of the same type. So to conclude, HPLC is an important diagnostic tool. However, some entities cannot be differentiated based solely on HPLC values. So this emphasizes the need for molecular diagnosis and genotyping the patient. Molecular confirmation may have importance in prenatal diagnosis and genetic counseling. Homozygous and compound heterozygous conditions for large deletions have high HbF values, which may be misdiagnosed as homozygous beta thalassemia. And in such situations, parental screening is mandatory to reach an accurate diagnosis. Thank you.
Thank you. Well presented. I just have one query. Your study was 16 years, of duration of 16 years.
Yes.
So you have preserved all this material. How did you retrieve it?
So, ma'am, we had all the reports in our database. I had to go to my database and take all the, screen all the patients of delta beta and Lepore and HPFH3 cases.
Questions? Don't seem to be any questions, so we'll go to the next.
Thank you, ma'am.
The next presenter is Dr. Preeti Tripathi, and she's presenting the utility of labile plasma iron assay in thalassemia major patients.
Good afternoon. I shall be presenting the findings of my study titled Utility of Labile Plasma Iron Assay in Thalassemia Major Patients. And this study was done at Department of Hematology at AIIMS, New Delhi. So to introduce, as we all know, LPI and labile plasma iron assay has been in research for almost a decade now. It is basically a redox active component of non-transferrin bound iron, which is potentially toxic once it enters the cell. The good part is that since it is redox active, it is easily chelatable also. So LPI has been mostly studied in the thalassemia major patients because iron overload is inevitable in these patients. And as we already know that ferritin, which is the most widely used marker as a marker of iron overload, has its own limitations and fallacies. LPI has been in exploration in the past studies as a marker of iron overload, as a measure of assessing the chelation efficiency, and as surrogate marker for early cardiac damage. So we started out to explore the same parameters in our Indian clinical settings in the study. So our study was a prospective study done over a period of 19 months. A total of 35 patients of thalassemia major patients were studied under two main groups. Group A were the transfusion-dependent thalassemia major patients who were not yet on any form of chelation, and Group B was the transfusion-dependent thalassemia patients who were on some form of regular oral chelation therapy. We also studied the 10 age and sex-matched healthy controls. So we followed these patients for a period of 1 year, and their ferritin and LPI assessment was done at baseline, 6 months and 12 months. For patients on chelation, that is the Group B patients, we had a paired LPI measurements, one before the chelator and one is 2 hours after chelator. These patients were instructed to withhold their morning dose of chelator till the sampling was done. Their clinical profile and other investigations were taken from records. We followed all the ethical guidelines. The serum ferritin was done using sandwich ELISA technique, and LPI was done using the Firos LPI kit, which is marketed by FRX Limited, by using the fluorescent quenching method on a multiplate fluorometer. The serum was collected and stored at -70°C to do the batch-wise processing. Positive and negative were defined as per the recommendations of the kit, and statistical analysis was done using SPSS version 21. Coming to the result, this is the table showing the characteristic of the patients in two groups, study groups. We had 13 patients in Group A and 22 patients in Group B. As we can see here, the patients in Group B had a higher mean age, had more number of patients with splenomegaly, a larger size of the spleen, and more patients with deranged LFT, but this is understandable because of the longer duration of the disease in these patients. Coming to the mean LPI levels, at baseline, we found the mean LPI levels to be higher in the Group B patients as compared to the Group A patients, which in turn was significantly higher than the LPI levels in controls. LPI levels as described in the previous study in thalassemia population is as shown here in the table. As we can see, the previous studies have studied various subpopulations of thalassemia. So our study mostly closely resembled to the study done by Kabantik et al., and the levels are comparable. Coming to the correlation between the LPI and ferritin levels, in both the groups at all the 3 time points, we found the LPI to be correlating with ferritin levels. Now this has some mixed results in the past. A study done by Swart et al. In 2016 found LPI to be correlating with transferrin saturation, but they did not find any correlation with ferritin levels, while the study done by Kabantik et al. In 2009 found the LPI levels to be correlating with ferritin, but only in the early stages of disease. Now following the trend of LPI and ferritin over 1 year follow-up period, as we can see here, both the LPI and the ferritin showed a significant upward trend in the Group A patients, while in the Group B patients, LPI showed a significantly downward trend, while the trend of ferritin was not found to be significant when the patients were on continued chelation. So this is an important observation in our study, and this indicates that when the patients are on chelation, LPI may be a more suitable marker because it is more sensitive to the presence of chelators in the body, while ferritin may not give the true picture of iron load of the body. And this limitation of ferritin has already been validated clinically in the past by studies of Adam Kivitz and Davis et al. Now coming to the LPI levels post-chelator, we did not find the fall of LPI levels 2 hours after chelator to be significant at any of the 3 time points. This is in contrast to the findings by study of Zaninali et al. In 2009, wherein they demonstrated the fall of LPI to be significant 2 hours after the chelator was administered in their study group. They had followed a group of patients of thalassemia major over a period of 52 weeks, and they found the fall of LPI after the chelator admission to be significant, and they concluded that this fall of LPI level can be taken as an efficiency of chelator protocol, and it can be used as a marker of chelator efficiency. However, our study could not find any significance. When comparing the LPI levels with other parameters, it was found to be correlating with increasing age and splenomegaly. However, no correlation was found with the transferrin saturation and T2-star MRI levels. However, since this value was available only in a very small number of patients, we could not derive any fruitful conclusion over this. Now, lastly, coming to the limitations of the study, small sample size and a short follow-up period is definitely a limitation. Also, we could not do the daily pre- and post-chelator measurement of LPI unlike the previous study, which could have given a better picture of the efficiency of chelation. And availability of T2-star cardiac MRI would have definitely added some more information to the study. To conclude, there is no single ideal test to measure iron overload in thalassemia patients. LPI is a potential alternate to ferritin levels. It is better than ferritin to monitor the trend of tissue iron overload in late stages of the disease, especially when the patients are on chelation. And we did not find it to be useful in determining the chelation efficiency, at least in the short term. And we also could not derive any conclusion regarding its usefulness as a surrogate marker for cardiac damage. At the end, I would just like to submit that despite our limitations and whatever small number of samples we had, this study being the first study of LPI in India, it can be a useful stepping stone to the studies to come in future, and we never know, LPI may be the test of future. Thank you.
Thank you very much, Dr. Preeti. Very good presentation. So do you want to recommend this as the first test after stopping ferritin, or you want both of them to continue for a while together?
No, ma'am, it's too premature to say that because LPI testing is, as of now, it is not very standardized. There are multiple number of kits and the procedures are also very different. The kits also are not very standardized as of now. We have about 4 or 5 types of kits available. So there's still time for it to be the recommended test. As of now, ferritin stays, ma'am.
Okay. Thank you very much. Dr. Deepika.
Yes.
Ma'am, can you come to the mic, please? I don't think people can hear here.
As you claim that you would like to really recommend for this test, usually ferritin is an accepted one.
Yes, ma'am.
But it is not very satisfactory.
Yes.
So you should have the T2 MRI, which is the best way to know which organ is really there is iron overload. Should have been a good correlation.
Yes, ma'am.
And second is that you are telling that this has a good correlation with ferritin. So what is the advantage? And this is more expensive or it is less expensive? That consideration is also there.
Yes, ma'am. T2-star cardiac MRI is definitely the gold standard for assessing any cardiac damage in the patients, and LPI should have been correlated with T2-star cardiac MRI only. But because of the limitations of our center, since T2 cardiac MRI was not being done in all the patients of our OPDs, we could not correlate it with that. And since the clientele is also very poor, we could not advise them to go for it outside also. Coming to the second point, the ferritin point, definitely, ma'am, ferritin is, as of now, it is the standard test for iron overload. But in the long term, in the late stages of disease, when the patients are on chelation, only in that, maybe that population, LPI may have an edge over ferritin, where ferritin are known to be fallacious and it has its own limitations. Ma'am, as of now, the kits are not being marketed in India as of now. So I cannot tell you the cost.
Because one would like to go who are managing the cases of thalassemia.
Yes.
T2 MRI rather than doing this.
Yes, ma'am, definitely. It is the gold standard. As on today, it is the gold standard.
Thank you, Preeti.
Yeah, I have one question. For both the groups, beta thalassemia major
Yes.
were having the serum ferritin on a higher side. What were the serum ferritin levels for these patients? Both the groups.
So if I can show the slides here. Could you explain without the slide? Ma'am?
Can you explain without the slide?
Okay, sir, the ferritin levels in the group A, that is the patients who were newly diagnosed, their ferritin level was less than 500. They did not have higher ferritin levels because these patients were the very newly diagnosed. They were not yet started on any chelation. While the other group, the group B, definitely since they were already on chelation, their ferritin was definitely much more higher.
The point here is that if the ferritin levels were less than 500 in the group 1, so how can you claim it to be a test for iron overload? Because LPI does respond to chelation immediately. I mean, it's got a short half-life, so we cannot claim it to be an indicator of iron overload if the group 1 was having a serum ferritin of less than 500.
Sir, we included those chelation naive patients basically to study the LPI levels in those patients, chelation naive patients. We wanted to compare the LPI level in healthy controls as compared to the chelation naive patient and with the chelation patients on chelation. That is why we included that group also. Otherwise, we wanted to assess the LPI levels only in thalassemia major patients on chelation.
See,
I think we'll have to stop the discussion here because it will eat into the time for the next speaker. This is Ms. Tejasri, and she'll be talking on the first report of dysfunctional mechanosensitive piezo ion channel protein, piezo, associated with hereditary xerocytosis in India.
Good afternoon, everyone. Here is the topic of my today's talk entitled First Report of Dysfunctional Mechanosensitive Piezo Ion Channel Protein Associated with a Hereditary Xerocytosis in India. To begin with the introduction, hereditary xerocytosis is also popularly known as dehydrated hereditary stomatocytosis. It is a red cell membrane dehydration defect exhibiting the loss of cation, particularly potassium and water content. The primary cellular cause of hereditary xerocytosis is mutations in the piezo1 gene. The disorder has autosomal dominant inheritance. It is a rare disorder with a prevalence of approximately 1 in 50,000 individuals. Unlike other red cell membrane disorders, splenectomy in HX is contraindicated due to the increased risk of thromboembolic complications. HX is clinically, genetically, and biochemically heterogeneous in nature. The primary clinical findings are anemia ranging from mild to moderate level, reticulocytosis, hepatosplenomegaly, and hyperbilirubinemia. The prominent laboratory findings are increased MCHC and MCV, decreased osmotic fragility, and the presence of stomatocytes and or spirocytes on peripheral blood smear, which mostly gets unnoticed. Piezo1 gene. It is located on long arm of chromosome number 16. It contains 51 exons, and the protein encodes for 2521 amino acid. It is one of the largest ion channel in RBC membrane with 36 transmembrane domains. It is a mechanically activated ion channel conducting mainly monovalent cations. Approximately 25 missense piezo1 mutations associated with HX have been reported. No report of piezo1 mutations from Indian population till date. Here the schematic representation of a red blood cell membrane structure has been shown, showing the piezo1 protein embedded in the RBC membrane. 5 patients with chronic hemolytic anemia showing the symptoms of hepatosplenomegaly with iron overload of unexplained origin were referred to us. Hematological tests including peripheral blood smear examination, reticulocyte count, and CBC were performed. The causes for hemoglobinopathies, red cell enzymopathies, and membranopathies were also ruled out. For the further genetic characterization, the samples were sent for NGS. The in silico prediction analysis of the identified mutations was done by using bioinformatic tools such as Polyfin2 and many other. Structural function analysis was also done of the mutations by molecular modeling software using PyMoles, SwissPDB, and many other. Here the hematological and the biochemical data of the 5 HX patients has been shown. All the 5 patients are showing similar symptoms with a varying degree of all the 5 patients have anemia ranging from mild to moderate level. They are showing macrocytosis with a slightly increased MCHC level. Their osmotic fragility test is decreased. All the patients were dependent on blood transfusion. The clinical manifestations of patients included hepatosplenomegaly, reticulocytosis, and hyperbilirubinemia. Propensity to iron overload was seen in some patients. Talking about red cell enzymopathies, all patients show normal result except case 1, which show a defect or deficiency of pyruvate kinase. The eosin 5 maleimide test for red cell membrane defect show normal result. Few patients also show the presence of stomatocytes. Genetic analysis of the identified piezo1 mutations by NGS. NGS detected 6 mutations, out of which 4 are novel and 2 are previously reported. All these mutations are missense in nature, and they are mostly present in the carboxyl terminal domain of the protein, which is located near the pore-forming region of the ion channel. The pathogenicity of the mutations was also determined, and few of them were found to be likely pathogenic. In the left-hand side diagram, 3 mutations are listed which are located in the inner helix carboxyl terminal domain, whereas the other 2 mutations are located in the peripheral helix. Both these domains are located near the pore-forming region, and variation in either of these domains can lead to delayed ion inactivation and cation permeability, resulting in dehydration of cells. Here a 3D model of a wild and a mutant type human piezo1 gene in exon 21 at 972 position, changing amino acid arginine to histidine in the peripheral helix domain of the protein is shown. It causes changes in the conformation of the protein, leading to cation permeability and dehydrated cells. Discussion. NGS analysis revealed heterozygosity for 4 novel missense mutation and 2 previously reported mutation in the piezo1 gene. Mutations were primarily found in the highly conserved carboxyl terminal pore-forming region of the ion channel. These reports have shown HX-associated piezo1 variants exhibit a gain-of-function phenotype, indicating that increased cation permeability may lead to dehydration of a piezo1 mutant HX erythrocytes. Piezo1 variants co-inherited with other congenital hemolytic anemias may ameliorate the clinical phenotype, as seen in case 1, who has HX in combination with heterozygous variant in PKLR gene, resulting in severe clinical manifestations with phenotypic variability. To conclude, the association of mutations in the mechanosensitive piezoionic channels of the piezo1 gene as the primary cause of HX has helped us to understand the genotype-phenotype correlation. Until genetic diagnosis by NGS analysis was obtained, these HX patients has remained undiagnosed since birth, requiring regular transfusions. These results underline the importance of precise diagnosis in HX, particularly in view of splenectomy because of the increased thrombotic risk. The rise of next-generation sequencing provided a sensitive, cost-effective, and rapid tool for understanding the molecular pathology of undiagnosed cases of congenital hemolytic anemia. Acknowledgements, I would like to acknowledge Dr. Prabhakar Kedav, Dr. Manisha Madgaikar, and all the other doctors from whom these cases were referred to us. Thank you.
How did you suspect them as possible hereditary xerocytosis before you did all this?
It has been reported in the literature, all the symptoms, hepatosplenomegaly, macrocytosis with increased MCHC, and iron overload with the presence of spirocytes or either stomatocytes can lead to hereditary xerocytosis. And by doing eosin 5 maleimide test, it was clear that it is not a case of hereditary spirocytosis. So for genetic characterization, we thought we should send the sample for NGS so that we get to know the defect at molecular level. It can help in diagnosis for the future generations also.
Any other questions? Sorry, ma'am? Madam is asking the mode of inheritance.
Autosomal dominant. Dominant.
But have you studied the family?
They were not showing symptoms.
So it will be difficult to comment on that because none of these have been studied, the family pattern.
No.
That would have been nice to know. Second is that what is the follow-up of these cases?
Sorry, ma'am?
Follow-up of these cases. Hemolytic anemia, if it is there, what is...
They were showing hemolytic picture, but...
No, but other causes has to be also excluded.
Hemoglobinopathies, enzymopathies and membranopathies were performed and they all were showing normal results. Thank you.
How many cases did you screen to get these 5 cases positive?
We come across many cases and these 5 cases were showing similar symptoms. Basically these 5 cases. So to find the defect, we thought of sending them to NGS and we got the same defect in each one of them.
So basically you sent 5 and all 5 were stomatocytosis.
No, we have a funded project and we send
Other
many
mutations
cases.
were also
Yeah.
seen. Okay.
So what is the pathognomic finding for automated blood cell counts with hereditary xerocytosis?
Sorry, ma'am?
What does the automated blood cell counts show? What are the red cell indices in a typical case of
Basically,
HX?
they have said that we get to set macrocytes, but basically we don't get to set microcytes.
So is it macrocytosis which is pathognomic or something else?
I think stomatocytes which are...
No. Okay. Let's move on to the next
Thank you.
case. This is going to be presented by Dr. Rucha, and she's going to talk on simple APTT-based tests for differential diagnosis of hemophilia A and B, preliminary results.
Good afternoon. Myself, Dr. Rucha Patil from National Institute of Immunohematology, Mumbai. Today, I'll be presenting the study entitled Simple APTT-Based Test for Differential Diagnosis of Hemophilia A and B, the preliminary results. So as we know, hemophilia is an X-linked genetic disorder wherein the patients tend to bleed excessively. India stands second in number of hemophilia patients in the world according to the World Federation of Hemophilia. However, there are only approximately 21,000 of our patients registered with the Indian Hemophilia Registry, whereas the expected number is 85,000 to 1 lakh. Why are so many patients undiagnosed? There are many reasons. For example, the unawareness among the patients, among the clinicians in remote areas. One cause is also there are only a handful of comprehensive diagnostic centers for hemophilia in India. What we have observed is that there are many laboratories which perform the basic coagulation screening, that is prothrombin time and activated partial thromboplastin time. But stop there. They do not do the factor assays which determine the factor 8 and factor 9 levels, which can help differentiate the hemophilia A and hemophilia B patients. This non-availability may be due to the non-availability of expertise or the instruments like -80-degree freezers, which are required for the storage of reagents for these assays. This, however, the non-availability of this factor assay is a major drawback. Thus, the main aim of our study was to standardize and establish a rapid, simple, and cost-effective screening APTT assay, which will at least give us an idea whether the patient is factor 8 or factor 9 deficient, wherein all the reagents can be stored at 4 degrees Celsius refrigerator itself. And we achieved this by using factor 8 and factor 9 coated beads. So the principle of the test is same as the APTT, wherein a activator like kaolin is used, which triggers the intrinsic coagulation cascade by activating the contact factors, which in presence of phospholipids and calcium leads to thrombin generation and fibrin clot formation. Now the difference here is we add one more reagent, which is the buffer-coated beads or the factor-coated beads, and this is nothing but a modified correction study. So when you add factor 8 beads to a hemophilia A patient who has prolonged APTT, this prolonged APTT will get corrected. Similarly, this correction will not be seen in the plasma of a hemophilia B patient. Now, when you add factor 9 coated beads, the prolonged APTT of a hemophilia B patient will be corrected, but this correction won't be seen in a hemophilia A patient. So this is the principle of the test. Now, before recruiting the patient, all the conventional tests were done so as to diagnose the patients to hemophilia A, hemophilia B, and von Willebrand disease. Now, the main task in hand was making of these factor 8 and factor 9 coupled microspheres. For this, different factor 8 and factor 9 concentrates were used, both plasma-derived and recombinant, and the concentrations used were from 200 to 1,000 units, and they were coupled to 2 micrometer polystyrene beads by passive adsorption using different protocols. The protocol which we use is as shown in this slide. Now, the final solution was aliquoted and stored at 4 degrees Celsius, and the stability of these beads were checked after every 15 days. This was done using FITC-labeled anti-human factor 8 and factor 9 antibodies using flow cytometry, wherein the mean fluorescence intensity is recorded and the coefficient of variation is calculated. Along with this, we even did correction studies using factor 8 and factor 9 deficient plasma. So this is the workflow of the study, wherein total 50 severe hemophilia A patients were recruited, along with 20 hemophilia B and 30 healthy donors, and this modified APTT test was performed on these samples. Now, out of the 50 hemophilia A patients, 16 were inhibitor positive. However, there was no significant difference seen in this correction studies in inhibitor positive and negative samples. So coming to the main results, now, as seen on table 2, the mean correction which we found using plasma-derived factor-coated beads is noted. We got excellent correction using plasma-derived factor 8 coated beads of 27.1 and 28.9 seconds in hemophilia A patients with and without inhibitors. And this correction was not seen after we add factor 9 coated beads. Similarly, the correction of 45.5 seconds was seen when we add factor 9 coated beads to hemophilia B patient, which was not seen on the addition of factor 8 coated beads. Now, the correction using factor 9 coated beads is not as good as factor 8, but it still helps us differentiate between hemophilia A and hemophilia B. Now, along with these patients, we even took 2 VWD patients wherein the factor 8 levels were lower, that is 8%. We found that factor 8 coated beads correct even the von Willebrand disease patients' APTT. Now, coming to the mean correction observed using recombinant factor-coated beads, here the correction found using factor 8-coated beads was 35.8 and 37.1. However, using factor 9-coated beads, there was no correction seen in hemophilia B, showing that the passive absorption of recombinant factor 9 was unsuccessful. Now, coming to the bead stability, the plasma-derived factor 8 beads, which was found to show excellent correction, had a stability of 6 months, which you can see in the below table, wherein it was after 6 months that the correction reduced. Now, the recombinant factor 8 beads showed a stability of 1 to 2 months. Plasma-derived factor 9 beads showed a stability of 1 month, and recombinant factor 9 was not properly absorbed. So the best result seen was in plasma-derived factor 8 for both correction and stability. However, the next step is to try more different modifications so as to increase the stability and binding. Thus, it is a dilemma for treating clinicians and patients when diagnosis for hemophilia cannot be given, especially in emergency situation. The above modified APTT assay is rapid, simple, and cost-effective. It gives us an idea whether the patient is factor 8 or factor 9 deficient, wherein the reagents could be stored at 4 degrees Celsius itself. Once all the beads are made with proper stability, the approximate cost of this test for one patient using all the 3 beads would be around only ₹95. The preliminary results show that the test is indeed useful. However, the bead stability needs to be improved. The limitation of this test is that it cannot distinguish between hemophilia A patients and von Willebrand. However, in countries like India, where many patients are still left undiagnosed, such simple, inexpensive tests could help diagnose majority of our patients all over the country. A detailed and accurate diagnosis can thereafter be done. Thus, if made available in different parts of the country wherein no major instruments or freezers are available, at least the initial diagnosis and thus management could be possible, and this could help reduce the mortality which happens during major hemostatic challenges in undiagnosed new patients. Thank you.
Seems to be a very cost-effective and good idea. These making of these beads, was it very technically difficult, or you all standardized it? I mean,
no, it was. We had to use n number of buffers, the pH, etc. So that is why still we have to do more work with that, but it was technically difficult.
Any questions?
You know that the diagnosis of hemophilia A and B, it constitutes a mixing studies wherein normal plasma is mixed with the patient plasma.
Yes.
And the correction takes place. Did you compare your findings with the mixing studies which are available?
Yes, yes, the normal NPP mixing test, right?
Yeah. So,
yes, yes.
So was there any difference? Because if you are going to standardize this kind of procedure,
it should be according to the NPP.
There should not be a significant difference between the mixing studies and your, bead analysis.
With the factor 8 beads, we found the correction very similar to the NPP mixing studies, but with the factor 9, we have to yet improve over that.
Thank you.
I have one comment and one suggestion. Why don't you make it a commercial product, transfer it to a commercial person, and make it like a kit? This is the suggestion. Before that, I think it's a good idea to see the microparticles or the polystyrene which you are using in the electron microscope. How are they distributed and what they are absorbing. So at different stages you can have that. This is my suggestion. I think it's a very good study. It's an innovative one.
Thank you. We'll go to the last speaker, Dr. Madhavi Madali, a long amplicon PCR next generation sequencing method for efficient and co-effective molecular diagnosis of hemophilia A and hemophilia B.
Good afternoon, I'm Dr. Madhvi from the Department of Hematology, CMC Vellore. I'm here to present a study on long amplicon based NGS in the diagnosis of hemophilia. The accurate and timely detection of disease-causing mutations in patients with inherited diseases plays a pivotal role in this era of personalized medicine. This not only confirms the clinical diagnosis but also helps in genetic counseling and prenatal testing, also in correlating genotype with the disease phenotype. Hemophilia A and B are the two most common inherited bleeding disorders, where more than 55% of hemophilia A and majority of hemophilia B are caused by point mutations in factor 8 and factor 9 respectively. The conventional methods to identify these point mutations include both indirect and direct methods like linkage analysis, methods such as CSG, TTG, etc., followed by Sanger sequencing or direct Sanger sequencing. However, these methods are expensive, labor-intensive, and time-consuming for a routine diagnosis of hemophilia. Advancements in the field of NGS has accelerated the genetic diagnosis of several diseases. The many approaches of NGS include whole genome sequencing, exome sequencing, and targeted approaches such as a panel approach or amplicon-based methods. Among these, targeted NGS efficiently detects disease-causing regions of the human genome and sensitively identifies disease-causing variants. An innovative approach of integrating long PCR along with NGS has been used previously to map whole genome and recently as a targeted approach in identifying mutations in BRCA1, BRCA2 genes and TKD mutations, etc. With this background, we aim to establish a high-throughput long PCR NGS method for the rapid and sensitive molecular diagnosis of hemophilia. This study constituted 15 patients recruited prospectively from January to July 2018. The clinical diagnosis was based on the phenotype and factor levels. Among these, 10 patients were intron 22 inversion and intron 1 inversion negative hemophilia A cases, and the remaining 5 were hemophilia B patients. Both the conventional Sanger sequencing and the novel long amplicon NGS was done in both the groups. And the methods were compared in terms of the cost and turnaround time. In the next couple of slides, I would like to show the conventional approach done in hemophilia cases. This represents the flow algorithm that we follow in hemophilia A. First is intron 22 inversion PCR, followed by intron 1 inversion PCR. This is, once it is negative, either a multiplex PCR with CSG and sequencing is done, or a direct Sanger sequencing for all the 26 coding exons is done. Likewise, in factor 9, a multiplex PCR and CSG with Sanger sequencing or a direct Sanger sequencing for all the 8 coding regions is done. In our study, we have done direct sequencing in both the groups. In the second novel method that we have applied, that is the long amplicon method, the coding regions of factor 8 were covered in 9 fragments and factor 9 in 3 fragments. The amplicons were generated using multiple uniplex PCRs in a single program. The amplicons were then pooled and purified, followed by library preparation and sequencing. The bioinformatics of raw data generated by sequencing was analyzed by in-house pipeline, and we used a web-based software for annotation. This is a representation on your right. You can see factor 8, or the coding region is covered in 9 fragments. This is a gel amplification view of all the fragments which have nicely amplified, and a table showing the range of the fragments. In factor 9, it is covered in 3. The amplification of all the 5 patients is shown very clearly, and the range of the fragments. This slide is only to highlight that it is important to get robust amplification here, since the pooling of fragments is based on this. This is a table showing all the mutations identified in 14 of the 15 patients. In one patient, mutation was not identified by both Sanger and NGS method. It shows that NGS could detect a spectrum of mutations like duplication, deletion as large as 11 base pairs, and missense mutation. To your right, you can see an electropherogram representation of a patient with 4 base pair deletion by Sanger method, and the same patient analyzed by NGS to your left. This is an integrated genomic viewer representation. Lab NGS could detect all the mutation, and there was concordance in the result. In terms of efficiency, that is the turnaround time, the first 2 steps, amplicon generation, pooling, required only 6 to 8 hours. Library preparation till raw data was 5 days, and a couple of hours for bioinformatic analysis. As cost per sample, the pooled amplicons costed only 3,000 rupees. This shows that lab NGS required 1 week as compared to 4 weeks by Sanger for generating a result, and it was 4 times less expensive when compared to Sanger sequencing. This graph represents that it is a myth that NGS is always expensive, but when the number of base pairs to be sequenced is large, NGS is cost effective when compared to Sanger. Among the various NGS approaches, amplicon-based method is very cost effective, high throughput, and sensitive method. In conclusion, lab NGS was found to be highly reproducible in terms of identifying the mutations. It was superior to Sanger sequencing in terms of turnaround time, and it was less expensive. Our results demonstrate that it can be introduced as an accurate, rapid, reproducible test in molecular diagnosis of hemophilia. Also, the technique can be extrapolated onto other inherited diseases with multiple large genes. Limitations of my study are the less sample numbers, inter-lab validation to be done before it can be introduced as a diagnostic test, and the detection of large deletions and molecular mechanisms like inversion is still a challenge by NGS. Thank you.
Thank you very much, Dr. Madhvi. It was a very good presentation.
Thanks,
Any
ma'am.
questions, any doubts anybody has?
I can understand application of NGS for a large gene like factor 8.
Yes, ma'am.
Should we really be going for NGS in case of factor 9 where you have just 8 exons to sequence?
I wouldn't justify, but
I mean, it is like putting a complicated technique in front of us. And the number 2 is the turnaround time for Sanger sequencing is not 4 weeks, just 4 to 5 days.
Since we do a batch test and a lot of samples are run, so it would take that much time. This was what we found.
Thank you very much, Dr. Madhvi.
Thank you, ma'am.
I would like to congratulate all the speakers for excellent presentations and also sticking to the time. I think we have finished dot on time, which is a very good thing. A big round of applause for all of them.
Thank you, judges and speakers. Now we'll move on to the next session. The next session will be on pediatric disorders. I invite the chairpersons, Dr. Ramaswamy and Dr. Prashant Sharma, to come onto the stage to start the session.
Welcome you all. Without wasting our time, as we are in the right time, probably we would like to start in the timely fashion. And first of all, we would like to, glad to have Dr. Seiji Kojima, Emeritus Professor of Nagoya University, so to enlighten us on the talk on the refractory cytopenia of the childhood, first of the talk of the 4 talks, and mic is hand out to you, sir, please. And the credentials are a lot, but to sum up a little, I would like to say that he is a pioneer therapy for childhood aplastic anemia. He had been a chairperson for Japan Childhood Aplastic Anemia Association Society since 1992, and also had been the chairperson of Aplastic Anemia Working Party of the Asia Pacific Bone Marrow Transplantation Group since 2009. He authored over 395 publications in peer-reviewed journals with an overall impact of 2241 that sounds what is his credentials. Please.
Thank you, Chairperson. Kind introduction. Also, I am thanks the organizing member of this wonderful conference. It is a great pleasure and honor for me to talk about our experience of refractory cytopenia of childhood. Clinical and histopathological distinction between aplastic anemia and hypoplastic MDS is challenging. In addition, congenital bone marrow failure should be excluded from acquired bone marrow failure syndrome. The 2008 WHO classification proposed the provisional entity of RCC, refractory cytopenia of childhood. The spectrum of patients with RCC is wide. About 80% of the patients have the hypocellular bone marrow, like aplastic anemia, and the 20% normal and hypercellular bone marrow with dysplasia, meeting the criteria for RCMD. Aplastic anemia, complete lack of the hematopoiesis, and the RCCs are characterized by the residual patchy pattern of hematopoiesis, and RCMD is a normal and hypercellular bone marrow with a morphological dysplasia. In Japan, we established a central review system for bone marrow failure syndrome in children. Perihal blood and bone marrow smear were reviewed by two hematologists, and trypan push were reviewed by one hematopathologist, and PNH and telomere length were tested using the flow cytometry. And patients who suspect of the congenital bone marrow failure, we carried out the gene analysis. Between 2009 and 2016, total 1,712 patients were reviewed by using our central review system. Median age is 6 years, ranging from 0 to 39. Total 55% of the patients classified bone marrow failure. Among the 939 patients with bone marrow failure, 21 patients are classified as aplastic anemia, 41% RCC, and 22% RCMD, and 9% congenital bone marrow failure, including the Fanconi anemia, Schwarzman syndrome, and ischiosis congenita. We use the target gene sequence for the molecular diagnosis of congenital bone marrow failure syndrome. We can sequence more than 180 genes associated with the bone marrow failure syndrome. Among the 166 patients suspect of the congenital bone marrow failure, 50 patients, 30% of the patient has the phenotype, genotype, matched germline variant. However, 10 patients, 6% of the patient phenotype and genotype are mismatched germline variant. For example, UPN 219, clinically we diagnose the dyskeratosis congenita. However, molecular diagnosis reveals the Fanconi gene mutation. Correct diagnosis, this patient is a Fanconi anemia. Also, among the 181 patients with acquired bone marrow failure syndrome, we found the germline variant among the 10 patients, 6%. This is a so-called cryptic congenital bone marrow failure syndrome. 4 patients showed the Fanconi anemia associated gene mutation, and 3 patients the dyskeratosis congenita associated gene mutation. Historically, patients with hypocellular RCC are diagnosed as aplastic anemia in the majority of the countries. The proposal of the RCC from WHO remains controversial in the majority of countries, including Japan. So publication of RCC is limited to the member of EWG group, especially the publication from the United States and European country other than EWG group is very few. This is the opinion of the researcher in United States. RCC has been a controversial entity and has not been universally accepted, especially because the therapeutic and prognostic implications of the diagnosis have only been validated in a limited number of studies. So efforts are designed to further characterize the molecular understanding of aplastic anemia and RCC would aid in differentiating these entities. In Japan, we conduct the prospective study for patients with aplastic anemia using the immunosuppressive therapy with host ATG and cyclosporine, which provides a unique opportunity to compare of the long-term outcome between the children with aplastic anemia and RCC who receive the same immunosuppressive therapy with host ATG and cyclosporine. Among 186 patients, 22 patients are classified aplastic anemia, and 90 patients is RCC, and 34 patients are RCMD. This figure shows the response rate. Aplastic anemia 52%, and the RCC 59%, and the RCMD is 56%. No difference of the response rate among three groups. Also, the right figure shows the cumulative incidence of clonal evolution among three groups. Also, the no difference of the incidence of the clonal evolution among three groups. Since 2009, we prospectively compare the clinical outcome between the children with aplastic anemia and RCC who receive the immunosuppressive therapy with rapid ATG and cyclosporine. Total 252 patients are recruited in this prospective study. 25 patients are classified as aplastic anemia, and the 131, 52% RCC, and 58, 23% RCMD. A patient with aplastic anemia, 57% had the very severe disease, and only 80% the non-severe disease. On the contrary, patients with RCMD, only 5% of the very severe disease, and 74% is the non-severe disease. This shows the chromosome abnormality at the diagnosis. Aplastic anemia is only one of 63 patients, 1%, and RCC is only 3 of 131, 2%. There is no difference of the incidence of the chromosome abnormality between aplastic anemia and RCC. Contrary, 9 of 58, 15% showed the chromosome abnormality in patients with RCMD, mainly trisomy 8. Next, we compare the somatic mutation using the whole exome analysis by NGS. As you can see, 4 of 41, 10% of aplastic anemia showed the somatic mutation. Also, the RCC patient, 6 of 59, 10% showed the somatic mutation, mainly because so the incidence and variety of the somatic mutations are totally the same among the aplastic anemia and RCC. On the contrary, 6 of 26 patients with RCMD shows the somatic mutation. Somatic mutation, including the MDS-associated gene, such as TP53 and the ZBP1. This shows the response rate to rapid ATG and cyclosporine. Aplastic anemia, 50%, and RCC 63%, and RCMD 64%. So the failure-free survival and overall survival also the same among three groups. So the strict separation of the aplastic anemia from RCC could not predict the clinical outcome. According to the WHO classification, aplastic anemia is separate from RCC, including the RCMD. However, we propose aplastic anemia and RCC should not separate each other. On the contrary, RCMD should separate from the RCC and aplastic anemia. This is our proposal. This is a treatment algorithm for children with aplastic anemia and RCC. In Japan, we use the same strategy for children with aplastic anemia and RCC. A patient with HL-matched related donor, BMT, is indicated, and the patient without HL-matched sibling donor, immunosuppressive therapy with ATG and cyclosporine is indicated. Everyone knows randomized study from NIH, United States, showed the inferior outcome of the patient who receives the rabbit ATG compared to the horse ATG. However, horse ATG is not available in the majority of the Asian countries, including Japan. Only rabbit ATG is available in our country. So we conduct the prospective randomized multi-center study comparing the different dose of rabbit ATG in patients with severe aplastic anemia in collaboration with China, Korea, and Japan. Total 258 patients were recruited in this prospective study. The result of the total cohort is planned to present the ASH meeting this year. In Japan, we measured the rabbit ATG plasma level using the ELISA kit on day 14 and day 28. Flow cytometry was used to assess the lymphocyte subset on each time point, CD3, CD4, CD8, and regular T cell. This is a patient characteristics of Japanese cohort. Patient number is 81. Median age is 13, ranging from 1 to 67 years old. 42 patients belong to the 2.5 milligram group, and the 37 is a 3.5 milligram group. Every parameter is comparable between the two dose group, 2.5 and 3.5. There is no significant difference of immunosuppressive response between the 2.5 and 3.5 dose group. The ATG blood level is greatly varied among both 2.5 and 3.5 milligram blue, from 0 to 100, very widely distributed. Also, the median ATG level is comparable between the 2.5 and 3.5 milligram on day 14 and day 28. Then we studied the correlation between the ATG level and the absolute lymphocyte count before the infusion of the ATG infusion and the patient body weight. Spellman rank correlation coefficient shows the negative correlation between the ATG blood level and lymphocyte count, and positive correlation between the patient body weight and ATG blood level. So we compare the kinetics of lymphocyte subset between the two dose group, 2.5 and 3.5. As you can see, absolute number and the lymphocyte subtype is totally the same between the two dose group, 2.5 and 3.5. So next, we differentiate the two groups according to the blood level. As you can see, the blood level, high-level group is the recovery of the lymphocyte is significantly delayed compared to the low-level group, CD3, CD8, CD4 regulatory T cell at each time point. So this is the most important point. Response group showed the higher ATG level than non-response group at day 28. ROC curve we use to determine the cutoff level of the response and not response group. As you can see, cutoff level on day 14 is 21 microgram per milliliter. And the cutoff level on day 28 is 4.8. A patient whose ATG concentration is more than cutoff level, 92% respond to rabbit ATG and cyclosporin. Also, the cutoff level more than 4.8 at day 28. More than 80% of the patient respond to rabbit ATG and the cyclosporin. Every report from the NIH shows the rabbit ATG response rate is less than 40%, quite different from our data. Also, the patient who develops the EBV reactivation shows the higher ATG level at day 28. However, no patient develops the clinical EBV LPT. So also I check the ROC curve for detecting the day 28 ATG cutoff level to discriminate the positive and negative EBV reactivation. Cutoff level is 12 micro per minute. Multivariate analysis identified the severity of the disease and the ATG level at day 28 as an independent unfavorable predictor to respond to ISCT at 6 months. So our data shows the optimal level of ATG concentration at day 28 is 5 to 10 microgram per milliliter. So in the future clinical trial, I propose one arm is individualized ATG dose according to the optimal ATG concentration. Another arm is fixed ATG dose. Randomization of the two arms is very interesting and warranted. Next is bone marrow transplantation topics. Current standard condition regimen for aplastic anemia and RCC is mature-related donor transplantation is CY, cyclophosphamide 200 mg per kilogram plus ATG, and unrelated setting is fludarabine, half-dose CY, ATG with or without loss of TBI. However, we frequently experience the donor-type aperture, which means bone marrow failure with donor chimerism after HSCT. So risk factor of donor-type aperture is morphological classification, namely RCC is one risk factor of donor-type aperture. So we searched the risk factor of this complication. A total 660 children with aplastic anemia received the bone marrow transplantation between 1980 and 2010 in Japan. Patient who condition with fludarabine-based regimen, 220 patients belong to this group. The this complication incidence is 12%. On the other hand, non-fludarabine regimen, 440 patients is 3%, significantly higher than fludarabine regimen. However, next, we search the impact of the cyclophosphamide dose, because in Japan, we reduce the half dose from the start of the 200. As you can see, Fludara CY 200 mg group is no patient develops this complication. On the other hand, full CY 100 is 15% experience this complication. So what is the optimal conditioning regimen for acquired bone marrow failure children with high risk of donor type aplasia, namely RCC patient? We suppose the more intensification of the myelosuppression may be necessary. Fludarabine, melphalan is more myelosuppressive compared to the cyclophosphamide. So we compare the two regimens, full male base regimen and full CY regimen for aplastic anemia. Total 603 children with acquired bone marrow failure received the HSCT in Japan, and the 36 patient condition full male regimen, and 270 is a full CY regimen. As you can see, the 5-year overall survival and 5-year failure-free survival is a perfect 100% in full male regimen group. On the other hand, full CY group is overall survival is 93%, and failure-free survival is 86%. 9 patients experience the secondary graft failure, and 12 patients experience the donor type aplasia. So we recommend the high risk of donor type aplasia. Bone marrow just before transplantation is not aplastic anemia, namely RCC. Mature BMT is fludarabine, melphalan, and ATG, and unrelated setting, fludarabine, melphalan, ATG with 3-grade TBI. I appreciate all the members of the JSHCT pediatric aplastic anemia working group and Japan Childhood Aplastic Anemia Study Group. Thank you for your attention.
Thank you, Professor Kojima. The talk is now open for questions and comments. So RCC is a very challenging diagnosis in India as well. And in fact, some of your studies which had more RCC patients than aplastic anemia patients would be very difficult to conduct here, because even if you do cytogenetics, the patients you can diagnose confidently as not being idiopathic or acquired aplastic anemia, but being refractory cytopenias would be very minuscule.
So as the same in Japan, the RCC is more high according to the report from the pathologist, but we don't care, neglect that diagnosis in Japan.
Thank you very much. I think there are no questions. I would like to now invite our second speaker, which is Dr. Shekhar Krishnan. Dr. Krishnan is a senior consultant of pediatric hematology at Tata Medical Center, Kolkata, and he's currently on secondment from the University of Malaya, where he's an associate professor. His clinical interests lie in benign and malignant hematology and stem cell transplantation, and he's a graduate from CMC Vellore, and subsequently did his MRCP in pediatrics in 1996, followed by FRC Path in 2009, and then PhD from the University of London in 2011. Dr. Krishnan, please.
Thank you very much for the introduction, and thank you all for the attention, and thank you to the Haematocon Scientific Committee and the Organizing Committee for the opportunity to deliver this talk and for the opportunity to participate in this meeting. I have 20 minutes, so I have to dive very quickly into this subject. It's on a condition of Philadelphia positive ALL, current strategies. Just some introductory remarks. Philadelphia ALL is a rare condition in pediatric ALL, 3 to 5%, older children. A proportion of them tend to have high white blood count, a higher proportion tend to have CNS disease, and typically it's characterized in the majority of patients by the isolation of the P190 or the minor transcript, BCR-ABL1 transcript. It has captured our imagination because of the opportunity for targeted therapy using the ABL1 tyrosine kinase group of inhibitors, and that is why there is so much excitement about this particular entity. Before we evaluate current strategies, I think it's important to look at the history of treatment. So in the pre-tyrosine kinase inhibitor era, the paradigm of management was intensive chemotherapy treated as high-risk disease. Remember, these are all B-cell progenitor or B-cell precursor ALL. So intensive chemotherapy on the high-risk arm of the chemotherapy protocol, followed mandatorily by allogeneic stem cell transplantation with the genotype match donor. Retrospectively, these are the outcomes. So I'll just quickly show you. So I'm not able to get it. So the point I wanted to make is that the outcomes that we see are about 30% overall, and retrospective analysis suggested that you can stratify the outcomes based on presentation characteristics. These are looking at age and white blood cell count at presentation. So patients who are younger, low white blood count seem to do best with this approach, and but the outcomes vary anywhere between as low as 20, as good as 50. And that is the same point being made here. What I'm not saying here, but I will say in the subsequent slide, is that analyses also showed that early response to therapy based on clearance of circulating blasts or early bone marrow remission is also a good outcome, and that paradigm has been taken forward. I'll come to that in a moment. So the subsequent family of studies were looking at the introduction of imatinib into the chemotherapy regimen, followed by allogeneic stem cell transplantation. So keeping the original approach, but adding imatinib into chemotherapy. And the trial that has looked at this in a large way is the European Philadelphia ALL group of the trial. This is a multi-center, European-based collaborative study, so it involves about 10 centers in Europe. And the initial study was looking at adding, so in 1 to 17 years, so just go through the data. This is the high-risk arm of treatment. Early response was used to characterize patients into good risk and poor risk groups. The imatinib, just to draw attention to this, is about 300 milligram per square meter a day, post-consolidation, so after induction, discontinuous regimen, I'll come to that in a moment, at least 10 weeks before transplant or 18 weeks with chemotherapy, and those were the groups here. Transplant was mandatorily introduced here. That's the schema of this protocol, so just the randomization with imatinib or without imatinib, and that's the discontinuous schedule, right up to the intensive phase of chemotherapy and not in the maintenance phase. Now, the outcomes are quite impressive, especially because in patients with poor risk disease, you can see that the outcomes have now, in all patients, 60%, in poor risk group, 50%. Remember, the best outcomes in the non-imatinib era was 50%. So poor risk patients are now 50%. With imatinib treatment, about 70%. But the question really is, did imatinib make a difference in the good risk group, the randomization group? Clinically, you would say that that is significant. So patients who had imatinib seem to do better than, patients who had imatinib seem to do, I apologize, I think I have made a mistake here. So patients who had imatinib were actually better in terms of outcomes, both statistically, that could not be demonstrated. On an as-treated analysis, you can see, but on the intention to treat, there was no real response. So the point I'm trying to make is that perhaps the impact of the good risk patients is not so much the imatinib, but improvement in supportive care and perhaps better transplant outcomes. So just a point to think about. This is a very complicated slide, but the point I'm trying to make here is that MRD can be monitored in two ways. One is looking at the clonal immunoglobulin T-cell receptor gene rearrangements by quantitative PCR, or by monitoring the BCR-ABL1 transcript. And the question is, which approach is better? So if you look at this is induction, consolidation, and later time points using the immunoglobulin T-cell receptor gene rearrangement, and this is using the BCR-ABL1 transcript. And you can see there's a steady decline here compared to here where it tends to plateau, and sometimes it's a bit discordant. Now, this particular approach, this is a post-consolidation, seems to be discriminatory. So you can stratify patients. These are patients who have cleared at induction. These are patients who cleared at consolidation. So you can see clearly discriminatory, whereas here it is actually discordant. For instance, this particular red line refers to patients who are high positive, yet their outcomes seem to be. So discordant here, not very informative, it's more informative here. So the conclusions that we can take away from this particular approach is that introducing a tyrosine kinase inhibitor, an able tyrosine kinase inhibitor with chemotherapy followed by transplant, 20% outcome improvement, especially in poor risk disease. Poor risk disease as means patients who have poor early response to treatment. In good risk disease, effect is probably less obvious, and MRD monitoring perhaps best done using immunoglobulin T-cell receptor gene rearrangements, and then perhaps using flow MRD. BCR-ABL1 transcript monitoring perhaps not so informative, but I'll come to that in a later slide. Of course, the holy grail is, can we do away with transplant at all in patients in first remission? And that is the interesting question. Now, this is the pre-TKI era, and you have some interesting data here. If you look at this, these are patients pre-chemotherapy, pre-imatinib, with chemotherapy alone. You can see there is outcome in these patients. This is disease-free survival, it's overall survival. But what I wanted to draw your attention to is the fact that the outcomes tend to plateau very early in the transplant patients, so about 2 years, whereas in the chemotherapy-only treatment patients, you can see that there is a continuing decline in outcome and plateaus much later. And that's something that we'll come back to later in this point. So there is outcome in patients. So there are a group of patients who can be treated with chemotherapy alone. Who are those patients? Difficult to know. This is in the SFAL trial, the one that I talked about earlier, where imatinib was introduced with intensive chemotherapy and all patients were taken to transplant. You can see that there is a small proportion of patients, about 30 patients, who did not have transplant, and those are their outcomes. So roughly about, say, 40% of patients can be treated with imatinib chemotherapy alone, but who are those 40%? That is the important question. Now, a lot of excitement came in 2008 in the ASH-breaking abstract from the COG group based on this particular trial, the AALL0031, and this reported the use of imatinib in patients. This is introduced post-consolidation at that dose, very long, continuous chemotherapy, continuous imatinib with intensive chemotherapy. That is represented here in this black line. This is disease event-free survival, overall survival. And you can see that the outcomes in these patients, so these patients could not be transplanted because the genotype match donor was not available. So if you look at their outcomes compared to patients who went on to receive transplant, comparable. And that is represented here. Initially, when it was reported early in 2009, very impressive outcomes of 80%, but subsequently stabilized to around 70%. And so imatinib, there is a group of patients who can be treated with imatinib chemotherapy, and that was the message from this particular study. And that's the, so that's the cohort that you saw earlier, and that's the cohort post-consolidation treatment, continuous dosing with imatinib right through all intensive phase of chemotherapy, and perhaps pulsatile introduction in maintenance. That's the cohort we talked about just a while ago. Then the question, of course, is what about using a second-generation tyrosine kinase inhibitor? And there's a lot of rational reasons for using so. It's more potent. Dasatinib, for instance, is 300 times more potent than imatinib in terms of ABL1 inhibition. Number 2 is that it's CNS penetrating. And number 3 is that it is a broader kinase in terms of spectrum of activity. So perhaps it may be better. That is the intuition. And this is the data just published in 2018. The outcomes are probably no different using dasatinib in chemotherapy versus imatinib. So the point, I just highlighted here, and I just want to draw your attention to this particular column. And you can see here that about a third of patients, or maybe 40% of patients, are the ones who receive transplant, and a third to 40%. So nearly 2/3 of patients escape transplant in this particular group. Here, the rationale for using dasatinib alone chemotherapy, dasatinib alone without transplant, was based on early response, based on remission and end of consolidation MRD. That's the dose of dasatinib introduced in induction. And that is because of the Spanish data suggesting that early induction, better remission response. And clearly that's what's happening. If you can see, remission rates significantly improved, early MRD clearance better, but higher risk of infections. This is not easy treatment to deliver, and outcomes no different. So this is a point to be made that even though you get, so the interesting thing is that even though you get deeper response early, it doesn't translate into outcomes, and that's a point we'll talk about here. So based on the American data, the SFAL group went and gave imatinib for all patients in induction, and then transplant only the patients with poorest disease. And that's the data here. So I just draw your attention to this column. So that's the SFAL 2010. That's modified from this. This is the one that we talked earlier, which had transplant as the endpoint. And here, these are the numbers. Continues. This report is an abstract, and probably the manuscript is going to come out soon. Good and poor risk. Continuous imatinib in induction, and post-maintenance, in maintenance and also post-transplant, much better responses. Early remission rate, MRD clearance rates, but high risk of treatment-related mortality. So this is something I want to draw your attention to. Treating with imatinib is not trivial matter, but the outcomes are no different. So that's the point being made here. So even though in imatinib you can get away, your transplant rates are lower. I'll come to that in a moment. So look at this transplant rate, 38% versus 81%. Yeah? So the takeaway points from this, I just wanted to 5 points. So if you use end consolidation MRD as a criterion, nearly about 60% of patients perhaps can escape transplantation and can be treated with chemotherapy alone in first remission. Introducing TKI, better, deeper responses, somehow doesn't translate into improved outcomes. So just because you clear MRD doesn't mean, so MRD is not everything in the world of PH-ALL. MRD has to interact with the cytogenetic subgroup, and this is something that later yeah. So introducing TKI early in induction, not trivial. Infection toxicity is a big problem. Outcomes of dasatinib, still waiting on that, excess toxicity. And so the transplant role is now shifting into CR, not in CR1, in CR1 mainly for poor response patients, and moving more to a CR2 strategy. Just to highlight that as pediatricians, we must remember that there is a significant deceleration in growth with imatinib use, and that is over 30 months. You can see the standard deviation scores for height coming down. So how do we refine risk stratification? Who can we select? How can we select patients who require stem cell transplantation versus just chemotherapy alone? If looking at the pre-TKI era, it used to be mainly, I think, based on early response. You can see early response discriminates based on clearance of circulating blasts or early marrow response. In the SFAL group, presentation WBC count seems to show a poorer outcome. So patients with high presentation WBC count seem to do poorer with just an imatinib chemotherapy, even with transplant alone. They require different strategy. In the COG study and later in the SFAL study also, the molecular alteration Icarus is a lymphoid transcription factor in chromosome 7p12, and alterations of Icarus, typically deletions, seem to predict a group of patients who tend to do poorly. So this is another risk group. And of course, the question is, why doesn't MRD predict for outcomes in imatinib-treated patients who are given induction? This is maybe one reason for this, because there may be two types of PH-ALL. One is a CML-like biology, and one is a de novo PH-ALL. So this is in this particular publication. So this is PH-pod, the transcript only identified in ALL cells, but not in any of the other lineages, B, T, myeloids, and granulocytes, macrophages, granulocytes. So clearly, just looking at the lymphoid marker, you can say MRD clearance and patient does well. Whereas here, the BCR-ABL transcript is found in all lineages, and therefore, perhaps even when you clear MRD, this persists and therefore gives rise to relapse. So perhaps the CML-like biology requires a different treatment, but this is open for question, a speculative thinking. So what we can say is that in proposed management strategies is that intensive chemotherapy with imatinib is now the standard of care, but avoid hurry. So give it when counts are recovered, wait when the patient is clinically well. Remember, most of the COG-ALL data was post-induction outcome. Monitor response, maybe still not by BCR-ABL transcript. Allo-SCT, perhaps for these indications, the post-SCT imatinib still open. And there is now this trial, this collaborative trial going on in examining the use of imatinib with less intensive chemotherapy. Remember, there was a lot of mortality. Yeah? And using immunotherapy as additional strategy for patients with poor risk disease. So I think I have 5 minutes, so I'll talk about PH-like ALL, another entity that has captured a lot of imagination, mainly because of the opportunity for targeted therapy in that group of patients with ABL1 tyrosine kinase inhibitors. Now, the million-dollar question is, how do we identify these patients? The identification of PH-like ALL comes from the European and the American groups, one of them looking at gene expression analysis, which is different strategies. But in the diagnostic world, how do we do this? We have either a upfront strategy where we eliminate certain cytogenetic groups and just focus on these groups, or we can have a targeted response-based screening. Now, we are interested in this particular group because this is the one that responds to the tyrosine kinase inhibitors. This is the one which is associated with JAK2 kinase, JAK kinase alteration, so perhaps ruxolitinib. But again, this is just speculative. It's not in trial as yet. These are the ones that are of interest because can be potentially targeted with imatinib. And of course, Icarus alterations may be an indicator for these two, therefore it's used as a surrogate marker. So at the moment, we still do not know how best to recognize this. The next question is, even if you recognize it, does it make a difference? St. Jude data suggested perhaps no. So if you use an MRD-based strategy in the context of the St. Jude Total 15 very intensive chemotherapy protocol, no difference in outcome. That's a yellow line, a golden line, showing that no real difference in outcome. Yes, these patients have higher risk, these patients constitute a higher risk population, but if you use intensified therapy, no difference in outcome. Conversely, from the ANSCOG group, this is the Australia and New Zealand Children Oncology group, also working with the Dutch group, you can find that the PH-like ALL identified using gene fusions and the American approach of Tachman low-density arrays seem to define a poorer group who are, even using an MRD-based risk-stratified treatment, do poorly. So again, open question whether this actually makes a difference or not. So I think I've completed my talk in time. I just want to thank Professor Vaska Saha. He was my PhD supervisor, and also now I work with him at Tata Translational Cancer Research Center. I've been associated with him now for more than 15 years. And many of these ideas and understanding has been shaped by my association with him. I wanted to thank him and acknowledge him. And thank you for your patience and attention. Thank you very much.
Excellent session, sir. So, lucid presentation. Mic is over for the audience.
Hi, Shikhar. So, yeah, very comprehensive review of this complicated topic. So, although you have highlighted it in one of your slides, that post-SCT TKI maintenance, so there are no clear-cut guidelines. But whatever you have reviewed, whatever literature you have reviewed, any idea about how long it should be given, I mean, first of all, it should be given or not, and if it should be given, for how long it should be given?
So the weight of opinion is towards giving it post-transplant. The weight of opinion is giving it post-transplant. Many people wait for count recovery and normalization of kidney function. This takes about 12 to 16 weeks. And then if it's given, a minimum of 6 months is recommended. Some people give it for 12 months. Some people use a middle of 9 months. And they start very cautiously at 230 milligram per square meter, give it about 1 or 2 months, and then escalate slowly to the usual of 300 milligram per square. The main concern is the interaction with cyclosporin and other drugs which are used as part of transplant, especially the hepatic interactions, and because it's handled by the cytochrome P3A4 system. So that's the other concern. But the weight of opinion is towards post-transplant management.
Yeah, Mr. Krishnan. Excellent talk as usual. I have a practical situation for you.
Yes, sir.
We have a child, 6 years old, who has completed his protocol as per modified BFM 95 with imatinib from induction. We have managed all along very well. He is completed on 30th of September. Now, should I give imatinib post maintenance?
Okay.
With RQ-PCR negative?
So, sir, this is a very interesting question because there is, again, divided opinion on this. There is a group of people who believe that post-maintenance for about 12 months, BCR-ABL inhibition with TKI is advised, but the weight of opinion is, the answer is, the weight of opinion is not to do so.
No.
But my answer to you is that it is an area of uncertainty or lack of clarity. There is a group of people who want to do this. So the other point I wanted to make, so I hope I've answered your question,
Please.
sir. And I just wanted to add another point is that if I were a physician treating a patient and I have a child who has good early response to treatment with MRD clearance, if there is a matched sibling donor and there are resources that permit it and the donor and the patient is healthy, I may still be persuaded to give transplant a go, mainly because in the chemotherapy treated patient there is I didn't discuss, there is an incidence of continuing relapse, and even at 4 years follow-up that has not plateaued. So I just wanted to say that point.
It's the same thing I want to ask regarding how long we should continue imatinib. So you answered the question.
Yeah, so that is, so it is an area of uncertainty. Yeah. Yes, ma'am.
And how do you monitor with MRD? Each time you do RT-PCR, or how frequently one should follow the same guidelines like CML, or you have a
So, ma'am, for the pH-positive ALL treatment, the recommendation is to follow 2 time points. One is post-induction, and second is post-consolidation. And the hierarchy of testing is the qPCR MRD for the immunoglobulin T-cell receptor gene rearrangements, which are the clonal ones, followed by flow. BCR-ABL1 transcript gives discordant results, and sometimes it's difficult to know how to act, because BCR-ABL may persist, but not necessarily predict for
because
relapse.
this discordant feature we always see, MRD comes negative.
Yes.
But BCR-able transcript comes positive.
Correct, correct.
So which one to give?
So, ma'am, we use flow MRD, we use a qPCR MRD by T-cell, but if that is not available, flow-based MRD for the ALL, for the lymphoblasts. That's what we will use, ma'am. So at post-induction and post-consolidation. So post 2 time point monitoring, induction, consolidation. Remember, the consolidation time point is the one that is, predictive of outcome.
And you sort out the patient at that juncture for the need
Correct.
for continuing with chemo, imatinib alone versus HCT.
Absolutely right, correct. That's right.
I remain confused about the MRD because the German group still looking at, they say that look at BCR-able, not the flow.
Still use? German group still use?
BCR-able, every 3 months.
No, sir, I have a difference of opinion with you. I think based on the SFAL study, and now with the COG also coming, I think the weight of consensus is not to do the BCR-able transcript monitoring now. In fact, I would say that unless in a research setting, the BCR-able transcript monitoring is not the way to monitor MRD in pH-positive ALL. In pH-positive ALL, it is based on the lymphoid markers, either by TCR, T-cell receptor gene rearrangements or immunoglobulin, or flow-based MRD using...
No, I get your point, but I keep reading every day and then get confused about it. Only a couple of days ago, I read about the presentations at ASH 2017 by the German group and the ASH education book. They said that you better follow the BCR-able rather than flow.
So there is a divide between the adult and the pediatric world on this point, I think. I have a feeling. So I'm not sure whether this is pediatric literature that you're reading.
Why should it be different between pediatric and
it should not be different, but sometimes it should not be different, I agree with you. But sometimes all this evidence has come in pediatric literature, and there is a confusion about whether in adults we're looking at lymphoid blast crisis evolving out of CML, and whether that may be better monitored or better captured. So there is this. So this is an area of uncertainty. But as far as pediatric PH-ALL is concerned, the recommendation is to use ALL-based MRD monitoring.
So
excuse me. Regarding in the same context, the RT-PCR for the BCR-ABL is freely available. Easy can be done, whereas gene rearrangement study is a bit difficult. So the discordant result will arise only when there is a positive RT-PCR.
Yes.
Suppose if the RT-PCR is negative, so that is the clonality has been eradicated. So in that case, rather than going for the gene rearrangement, if it is a positivity, probably we need to look into that. If it is a RT-PCR is negative, the clonality is being eradicated, probably we can go by that further step based on that.
If the BCR-able transcript disappears and you're confident that the analysis is correct, but often cases we find, as I've shown you,
it is positive. Yeah. Majority of the time it is. I thoroughly agree. But in case it is negative, because we can extrapolate that.
Yes, you can potentially. Yes.
Hello, sir. In your practice, when do you introduce TKI in the induction phase? Okay. And how does it matter regarding the MRD assessment and also the risk of infection?
Yeah, thank you very
much. And what will be the average time period before the exposure to TKI during the induction before MRD assessment?
Yeah. So thank you very much. These are very important questions, and I would just like to thank you for the opportunity to answer this. So number one is that the thinking is that if you introduce imatinib in induction, the remission rates are better, the MRD responses are better. So as physicians, our motivation is to introduce imatinib in induction, and also for the families, I think this is a very important milestone. Nevertheless, the recommendation is to introduce it not to hurry with introduction. Even though day 15 is a recommendation, what we would do in the real world is to wait until count recovers and the patients are well. Now, for some patients, this count recovery may almost come at the end of induction, in which case then imatinib becomes a post-consolidation introduction. So the answer is that imatinib should be introduced very judiciously when the patient is well and the blood counts are safe, number one. If we are concerned about imatinib, we want to start it cautiously, we can start at a dose of 230 milligram per square meter per day and escalate to a minimum of 300 milligram per square meter based on the SFAL data. That is the recommendation. So have I answered your question, sir? The risk of infection is a problem. It is because it is quite myelosuppressive. Imatinib is quite, sorry, I apologize, it's quite immunosuppressive, and patients continue to have risk of infection even though they may not have myelosuppressive, myelosuppression may not be there. So that is something, risk of infections are much higher. Yeah. I hope I've answered your question, sir. Yeah.
Let this be the, excuse me, let this be the last question. Please go ahead.
Importance of TKI vis-a-vis any other agent in routine agent in the chemotherapy arm, okay, in SFL trial also. So would you like to introduce imatinib early and do away with the other agents like Dono or other things? So how will you rate these two strategies?
So this is a good point. So if you look at the SFAL 2010, which is now reported in abstract, there was high incidence of mortality, 14% when imatinib was introduced in induction, and 14% mortality, that's very high. And even the post-transplant mortality was 19%. So there is a randomized trial going on, which is investigating taking away the BFM HR blocks and replacing them with high-dose methotrexate at 5 grams per square meter. The data on that is still awaited. So I don't have information on this. But yes, this is things that people are exploring.
Thank you, Dr. Shekhar, for the valuable information for us. Okay. With this, we'll go on to the next third session. Meanwhile, I would like to say a word. All those who are judges for the poster session, please be requested to meet Dr. Ewens at 4 p.m. at the poster area. Please, reminder for all of the judges. Coming to the session continuation, I would like to invite Dr. Gaurav Karya. The talk is on the neutropenia in children approach to diagnosis. He's currently a consultant of pediatric hemato-oncology and bone marrow transplant at BLK Super Specialty Hospital in New Delhi. Instrumental in starting the pediatric hemato-oncology services in the same hospital since 2011. He played a key role in taking the bone marrow transplant program up to the next level in the pediatric transplant program. More than a decade of experience treating children as a BMT specialist. And he has treated children, both Indian as well as abroad patients. He has conducted more than 500 transplants so far for various disorders, including a thalassemia, sickle cell anemia, acquired aplastic anemia, Fanconi's, severe combined immunodeficiency, and so on. Thank you. Please go ahead.
Thank you very much, organizers, for having me here, for giving me this opportunity to interact with all of you. Just a slight correction. Thank you for the generous introduction. Just a small correction.
I'm sorry.
No, it's okay. So I've changed my place. So currently I'm working at Artemis Hospital, Gurgaon, another hospital in Delhi NCR. There are a number of hospitals. So, and it's a small world, so we keep on moving from one place to another. So I'm currently working at Artemis Hospital, Gurgaon, as a hemato-oncologist, pediatric hemato-oncologist and transplant physician from 1st January 2017. So the topic assigned to me today is very different from what Shekhar has already covered. So that was hardcore oncology, and now I'm jumping the gears and coming to very basics of hematology in pediatric practice. So the topic assigned to me today is approach to a neutropenic child in pediatric age group. So whenever we have any child or any disease with any particular symptom or sign, there are two basic objectives of evaluating that particular child. First of all, we look at the differentials, that what could be the possible differentials for that particular sign or symptom. And secondly, then we try to develop an algorithm or an evaluation approach to come to a particular diagnosis for that symptom or sign. Now, looking briefly at this pictorial, which shows how the neutrophils are synthesized. So we can see here that the precursor cells, the myeloblasts, they gradually mature into promyelocyte, myelocyte, and finally they form segmented neutrophils, which are mature neutrophils. And this is a gradual process. So any step, anything which happens in between might lead to defective production of neutrophils, either in a quantitative way or a qualitative way. Like the commonest thing which can be highlighted, we commonly read of severe congenital neutropenia, where there's a mutation, allele mutation or HAX1 mutation, where the defect is in the myelocyte. So we get a number of promyelocytes, and there is a maturation arrest at that stage. So based on where the defect is, we can have either the quantitative or a qualitative defect in the neutrophil function. Or so the overview of today's presentation will cover definition of leukopenia, neutropenia, the working definitions of neutropenia and leukopenia, key points in the history and physical examination, what should we look for, especially while evaluating a child, discuss the differential diagnosis of neutropenia, provide a framework for ordering the investigation, step 1, step 2 investigations, then briefly review the management of any particular child, a broad way of managing these children. So leukopenia, we know that the normal white blood cell count is between 4,500 to 11,500. However, when we say this, we should always have age matched controls in our mind, and we should always have it's a one-page chart which we should always keep in our OPD chamber, which shows that what are the aged related normal values for any particular child. For any child, and whenever we are assessing or interpreting a CBC, we should always compare it with the age match parameters. Whenever we are looking at the blood counts, we should emphasize more on the differential rather than the whole white blood cell count. And we should look what is the ANC, whether the ANC is appropriate for that particular age, whether the ALC, acute lymphocyte, absolute lymphocyte count, absolute neutrophil count is appropriate for that age or not. So coming to the definition of neutropenia, so the working definition of neutropenia is it is an absolute neutrophil count below the normal level by age and race is defined as neutropenia. The lowest limit of neutropenia in newborns is 6,000. So we can see how age or race affects the neutrophil neutropenia, definition of neutropenia. So in newborns, just born child, 6,000 per millimeter cube. Whereas if you evaluate the same thing after 2 weeks, it comes down to 1,000 per millimeter cube. Again, and this goes on till 1 year, all right? And beyond 1 year, it is 1,500. 1,500 per millimeter cube is the working definition of neutropenia. Looking at the differences which can arise because of the race. So in different races, there can be minor variations. So in blacks, the cutoff for neutropenia is 1,200 per millimeter cube. Now, after this, coming to the severity, and how does this severity affect the clinical presentation of any neutropenic child? So it can be divided into mild, moderate, or severe neutropenia, depending on what is the absolute neutrophil count. So 100 to 1,500 mild, 500 to 1,000 moderate, and less than 500 severe. And based on the neutrophil count, the clinical symptomatology can also vary from none to significant or repeated infections. The maximum risk of infection is when the absolute neutrophil count is less than 200, which is also called as very severe neutropenia. And the classical presentation in such cases is recurrent bacterial or fungal infections, and this is the hallmark of symptomatic neutropenia. We know how to calculate the absolute neutrophil count, so I'll skip this formula. Now, how to approach any such child. So basically, if any child presents to us with low white blood cell count and low neutrophil percentage, we should first of all see whether this is normal for this age or abnormal for this age. Then is that patient symptomatic as per the, and depending on the neutrophil, so if his neutrophil count is around 800, 700 or less than 500, is he having relevant symptoms also, and for how long those symptoms have been persisting. Whether it could be a congenital thing or acquired thing. So we have to take a relevant past history also, family history also. Transient, recurrent, cyclical, or persistent neutropenia, whether there could be some underlying malignant etiology or non-malignant etiology. Then coming to the marrow, whether there is a defect in the marrow or it is some kind of a peripheral process which is going on leading to neutropenia. Then decreased production versus increased destruction of the neutrophils in the periphery. So coming to history, just like any other pediatric disease, a very, very careful and elaborate history along with the extensive physical examination should be done, which is very relevant in congenital marrow failure states, of which congenital neutropenia is one small subcategory. So careful history taking, recurrent or unusual infections, bacteremia, pneumonia, cellulitis, sepsis, recurrent or recent fever which is persisting for more than expected time, any growth delays, any physical stigmata, aphthous ulcers, bone or extremity pain, rashes, abnormal or any abnormal history of, family history of neutropenia. While examination, we should always look for potential red flag signs like fever or ill-appearing child, any evidence of failure to thrive, dysmorphic features, lymphadenopathy, nodes more than 1 to 2 centimeters, so depending on whether it is supradiaphragmatic or above the diaphragm or below the diaphragm, so there are definitions of lymphadenopathy, so we should always look for all the lymph node groups, any periodontal disease, aphthous ulcers, oral lesions, any kind of skin abnormalities. Now this is extremely important because I'll tell you one incident where we had a young girl, 17, 18 years of age, presenting as aplastic anemia and very no failure to thrive, no other symptoms, no physical stigmata, regularly going to her school or college, and we were about to take her up for transplant, considering it is some kind of a drug-induced aplastic anemia, idiopathic aplastic anemia. And as a part of workup, we sent a comprehensive this marrow failure syndrome panel. And to our surprise, it turned out to be positive for dyskeratosis congenita. And then we went back and evaluated and re-examined that girl, and it was just one of her grade 2, which was dyskeratotic, and there were no other features of DKC. So although this is very uncommon, but this highlights the importance of an extensive and a thorough physical examination in any pediatric child being evaluated for either neutropenia or any other abnormal cell lines. Now coming to laboratory evaluation, a complete blood count along with a peripheral blood smear and a differential count. A comprehensive metabolic panel, liver functions, kidney function should be done. Immunoglobulin level assessment and anti-neutrophil antibody, although if it is negative, it does not rule out presence of immune-mediated neutropenia. Additional tests depending on what is the underlying diagnosis which you are considering. So for leukemia, you have to do other tests, bone marrow failure stoids. Apart from other tests, there are now very comprehensive NGS and whole exome sequencing based panels where you can do full assessment of your inherited marrow failure syndromes. For autoimmune, ANA is a screening, anti-dsDNA, C3, C4 level should be done. Viral titers, these are the most common viruses attributed, but in our scenario, we see leukopenia and neutropenia so common after dengue infections, okay? So viral panel should be done, and we should always keep in mind the current epidemic, viral epidemics which are going on in that particular region. So coming to decreased neutrophil production, so these are the primary etiologies: malignancies, aplastic anemia, congenital marrow failure states. Secondary causes can be infections or drug-related or immune-mediated or nutritional. Now, when we talk about the marrow, there is no defect in the marrow, so whether the platelet neutrophils are being consumed in the periphery, so it could be immune-mediated, drug-induced hypersplenism, or pseudoneutropenia. Now coming to differentials, what could be the possible etiologies? When the neutropenia is seen in neonatal age, the common etiologies are immune-mediated, so either it could be autoimmune or autoimmune neutropenia. Then it could be because of the placental insufficiency, PIH-induced neutropenia, congenital infections. TARCH is a very big group of neutropenia in neonatal period, and obviously infections like necrotizing enterocolitis. Infection-associated neutropenia, this is the commonest scenario which we see as pediatricians. So post-viral infections, leukopenia, and neutropenia, very, very common. Typical viral infections, EBV, CMV, RSV, influenza, HIV, and as I highlighted, routine infections which are endemic or epidemic in that particular area. It resolves in few months, multiple mechanisms, like it could cause direct suppression of the bone marrow, or it could cause neutropenia can be immune-mediated, or it could be because of the antibody-mediated damage. Now drugs, there are a number of drugs, so the extensive list is available of the drugs which can cause neutropenia. Chemotherapy we know, anticonvulsants we know, NSAIDs. This was something which I was not aware of, so rituximab, which we use very commonly nowadays for various disorders, it can also cause late onset neutropenia. So there are certain metabolic disorders which can cause neutropenia, like hyperglycemia, orotic aciduria, methylmalonic aciduria, and hyperglycemia, and more commonly glycogen storage disease type 1b. Primary immunodeficiency states can also lead to neutropenia. And a common, very classical example is in hyper-IgM syndrome, it is thought that the GCSF production, so hyper-IgM disorder is a B-cell disorder. One would debate that why will there be neutropenia in a B-cell disorder? But because of the CD40 ligand defect, there is defective production of GCSF, which in turn leads to neutropenia. So neutropenia can be there with B-cell defect, T-cell defect, NK cell defect, or as a combination of all of them. And whenever we are treating any such child, apart from treating the neutropenia symptomatically, we should always try and treat the patient, the primary disease, primary immune deficiency. Immune-mediated neutropenia, so the commonest is benign neutropenia of infancy. The incidence is 1 in 1 lakh. Duration is between 3 to 30 months. 90% of them recover without any significant problems. In adolescents or older age group, it can be because of other underlying autoimmune diseases. Now coming to congenital marrow failure state, so I will discuss, so it can be, as I highlighted earlier also, it can be either isolated neutrophil series, or it could be a part of the combination of the entire cell line. So when we talk about neutrophil series, there are three commonly encountered things: severe congenital neutropenia, which is also called as Costman syndrome when it is in autosomal recessive form. Another one is Sean-Diamond syndrome. So severe congenital neutropenia, it is a very variable presentation depending on what kind of underlying mutation is there. And you can see that this is, on the right side is the list of mutations which can be there in severe congenital neutropenia, and based on this, it can be divided into SCN1, 2, 3, 4, 5. Cyclical neutropenia, very interesting entity. So it is autosomal dominant. It is because of the same gene which causes severe congenital neutropenia, but the presentation is very benign and it leads to a cyclical pattern. The prevalence is 1 per million, really cyclical hematopoiesis, and the neutropenia can range from, the cycle can range from 14 days to 28 days. So it goes down and then it comes up. It goes down and it comes up. Neutropenia lasts for 3 to 5 days, and 10% of the patient develops significant life-threatening infections. And it can very nicely be managed once you know that what is the time when the neutropenia is going to peak. So if you introduce GCSF, they can be treated very easily without any significant problems. So this pictorial shows the difference of how the neutrophil peaks and goes to neutrophil peak and trough in severe congenital neutropenia as well as cyclical neutropenia. So in SCN, it is rock bottom all through, whereas in cyclical neutropenia, there is ups and downs. Diskeratosis congenita, we have talked about it. So this is another form of marrow failure states. People talk about Fanconi, but that is the classical prototype of marrow failure states, but we all know about it. So I thought diskeratosis congenita is something which should be discussed, and I also wanted to highlight it just because of the fact that we have encountered 1 or 2 cases where the presentation was very, very subtle of diskeratosis congenita. So this highlights the importance of a thorough physical examination. Then reticular dysgenesis, again, one form of inherited marrow failure states along with primary immunodeficiency state. So again, the neutropenia is very significant here. If we talk about the management of such cases, majority of these patients die unless they are intervened quite early in life with a hematopoietic stem cell transplant. And even doing a hematopoietic stem cell transplant in reticular dysgenesis is a very, very challenging task. WHIM syndrome, which is characterized by warts, hypogammaglobulinemia infections, myelocataxis. So again, here the neutropenia and excessive granulocyte apoptosis of WHIM syndrome are partially corrected by the GCSF or GM-CSF administration, and the immunoglobulins are also normalized with the administration of GCSF and GM-CSF. Coming to the management of neutropenia, depends on the clinical situation and underlying diagnosis. Often no specific management is required. Consider GCSF for certain diagnosis or recurrent infections. Follow-up is extremely important. Fever management, so fever, whenever there is a fever in neutropenic child, it should be managed as emergency. When we are dealing with the management of fever in a setting of neutropenia, it's largely dependent upon the age, etiology, severity of neutropenia and the history, past history. Get inputs from the hematology lab. Whenever there is fever and the neutrophils are less than 500, treat it as an emergency. Empirical broad spectrum antibiotics should be started after sending relevant investigations. Coming to an algorithmic approach after going through all this. So if we have a neutropenic child So first of all, the first question which comes into our mind is what were the previous neutrophils, okay? So whether they were normal or they were abnormal. So if the previous neutrophil counts were normal, so previous neutrophil counts normal, no, they were abnormal, so then it falls into the category of congenital neutropenia. That is how you will think about it. And if it has been normal, you should think about acquired causes. If we look at the approach to congenital neutropenia, so this is how the spectrum looks like. So if we talk about the combined immunodeficiency, so it will be CD40 ligand, Wilms syndrome, reticular dysgenesis, pancreatic insufficiency, Schwann-Simon syndrome, Pearson-Marrow syndrome. So this is a complete spectrum of inherited forms of neutropenia. When we talk about the acquired forms, so the commonly encountered are because of infections, as we have discussed. So infections are the commonest entity, with viral infections being most common, malignancies, malnutrition, so often because of B12 and folate deficiency, which is commonly seen in our adolescent girls, immune-related destruction, and other causes like drugs or other things. So this is a comprehensive assessment or analysis of evaluation of a neutropenic child. Thank you very much for our patient hearing, and I'm happy to take any questions.
Thank you very much for presentation in the timely fashion. So now there are questions which can be addressed.
Could you tell me how would a neonate, like born within first week of life with neutropenia, could be evaluated? And the emphasis is on like whether the torch test that people do diagnose it.
So how it can be evaluated?
In the first week.
First week,
Yeah.
okay. So basically, as I said, neutropenia in neonatal period, first week of life. So first of all, we should have the cutoff. The cutoffs are different in the immediate neonatal period. So whether the child is fitting or the neonate is fitting into that cutoff, and if yes, then we should obviously look at the clinical spectrum, whether there is any sign of infection like necrotizing enterocolitis, especially in the preterm neonates. If not, otherwise healthy looking child, no other suggestive criteria, then as you rightly highlighted, torch infection, torch workup should be done, and otherwise we should evaluate, look at the other if this is also negative, then whether it could be because of the immune-mediated, autoimmune or alloimmune causes. So these are the common causes of neutropenia in a neonate.
So you said Elaine mutations and neutrophil elastase mutations, they cause both severe congenital neutropenia as well as cyclical.
Yes.
So are they different mutations, or are there some other processes that drive them towards these different scenarios?
So basically, these are not different mutations, but somehow same mutation causes different kind of a presentation. So I mean, in cyclical neutropenia also you see this allele mutation, and in SCN also you see allele mutation. But somehow the clinical pattern is very, very different. So for that you'll have to observe a child on a regular basis. At one point of time, you can't say that no, if this mutation is positive, this strictly falls into SCN or it falls into a cyclical neutropenia.
Is there any role for giving a dose of steroid and then looking at the count after a few hours? That's generally mentioned when we evaluate children with congenital neutropenia. Do you find it useful?
So ma'am, you're talking about the neonatal neutropenia? In neonatal or?
Not neonatal, older children.
So older children, ma'am, role of steroids will be there whenever we are thinking of any immune-mediated etiology. Yeah. If we are thinking of a congenital marrow failure states, then steroids won't be of much help. So like if we talk about...
There's a test, like you give one dose of a steroid and then see the count after 6 hours.
Okay.
And there's something that if it responds, what it means. Did you find that useful?
It's basically... regarding your question, madam, when you are giving the steroid, it is going to push the neutrophil which are in the aggressed area from the endothelial cells, which is pushed into the circulation. Yeah. Half-life of the neutrophil generally from the bone marrow is 6 hours. Yeah. So that 6 hours it is going to there, but at the expense of chemotaxis. In a patient with a neutropenia, you are going to give a steroid. Chemotaxis is being tackled in the presence of already quantitative defect in the neutrophil count. You are going to increase this one. You will get a transient elevations of the neutrophils. Basically, the bone marrow as such in the neonate or in the pediatric, it will be less. Egress will be, but what you are pushing it by steroid is the pushing of the neutrophil which are in the endothelial. This one you are pushing it.
So it doesn't help us to differentiate between congenital neutropenia and cyclic neutropenia or anything like that.
Well, cyclic neutropenia probably it will not have that severity. Number one, it will have a periodontitis only the clinical picture. Most of the time it will be a self-limiting, and then it will have a presentation at the later age. Most of the time, if you see the cyclic neutropenia will be picked up at the age of adolescent as well as adult age group rather than this one. Whereas congenital severe neutropenia will be as onset from this one. This is the autosomal dominant inheritance, whereas the cyclic neutropenia. So there may be a family history of recurrent infection, periodontitis, and other things. Maybe that will give a clue to us.
One last question. So excellent talk, Gaurav. And in regards to management of these children, especially congenital neutropenia, what dose, how high we can go with GCSF doses?
Exactly.
And when would you recommend transplant in a child who's with a pediatrician who's managing with GCSF? Thank you.
So, sir, as far as the dose of GCSF is concerned, people have used doses of GCSF as high as 40 micrograms per kg per day to treat or to take them out of the bacterial infection. So, as high as 40 micrograms per kg has been used. As far as the indication or when to take them up for transplant is concerned, obviously it will depend on the clinical behavior of the child. So if a child has significant manifestations in the initial months of life, then obviously rather than subjecting or putting him at risk of any second infection which might be life-threatening, it will be reasonable to intervene early and to take him to transplant early.
Thank you very much, Dr. Gaurav, for the very exhaustive listing of causes and explaining how to approach them. Our last speaker for this session is Dr. Rahul Nethani, who's the senior consultant and the head of bone marrow and stem cell transplantation and clinical hematology at Max Healthcare in New Delhi. Dr. Nethani did his MD from Lady Hardinge Medical College, followed by DM from the All India Institute. He's a young and energetic clinical hematologist who's responsible for setting up the bone marrow transplant program at Max Healthcare. He's also a prolific author of research papers and has to his credit probably over 150 publications. And he's also performed more than 130 bone marrow transplants. So over to you, Dr. Rahul.
Thank you, Chairperson, for the kind introduction. Good afternoon, everyone. Today we are going to talk about a rare condition, a rare hematological condition, hemophagocytic lymphohistiocytosis. For a lot of pediatricians, it may be a tongue twister. So HLH is a better term to count. There was a time when it has 90% mortality. Unfortunately, today also it continues to have 90% plus mortality. And the idea about HLH diagnosis is not only knowing it yourself, the idea is also to educate your pediatrician and intensivists back home at your hospitals so that they suspect, they call you, and then only you can treat and have a better outcome. So whatever trials or other things that we may talk about that show fantastic outcomes, that can happen only if a child has been diagnosed in a timely manner, and that will happen only if such a diagnosis was suspected. I do not have any disclosures. So it's a hyperinflammatory syndrome, usually characterized by fever, hepatosplenomegaly, cytopenias, and some lab abnormalities. The classical finding that all of us have in our mind about this diagnosis is a hemophagocytosis, where a macrophage is engulfing either the erythrocytes or leukocytes or platelets or combination or their precursors. These are the incidences in different populations. So almost 100% of patients will have fever and cytopenia. Large majority of them will have liver dysfunction, which is actually more than hepatosplenomegaly. Many of them will have jaundice, coagulopathy. Some of them will have CNS symptoms at presentation. Lab findings, high ferritin is present in large majority, high LDH, and if you have ability to do the interferon gamma assays, they will be raised. Low fibrinogen, high triglycerides, and low NK cell activities, they also form the diagnostic criteria for this disease. Generally, it has been classified as primary or familial or genetic, or sometimes these are associated with immune deficiency states like Shidek, Higashi, Griscelli, X-linked proliferative syndrome. Usually, these children will be presenting less than 1 year of age and many times at 2 months or sooner than that. This is one of the child that we had with Griscelli syndrome, whom eventually we had to transplant because of his HLH. Hemophagocytic lymphohistiocytosis or HLH can also be secondary. And there are a host of reasons which can incite this process. Remember, it's an hyperinflammatory process. So any infection, any immunological insult can set up an inflammation, and then it can go out of control and you lead to HLH. So it's important to look for an underlying cause, because if you have a secondary HLH, treatment of primary disease is of utmost importance. How the immune system usually works is there is a trigger, there's an activation, and then there's cytotoxic or natural killer cell or cytotoxic T lymphocytes. There are different stages of maturation, polarization, docking, priming, fusion. Eventually, you form a pore into the target cell, release your granzymes, and the cell is killed. Now, what happens, and that is how different mutations in HLH have been thought of. The mutations in different levels are going to alter this process. This leads to uncontrolled expansion of T cell because the target cell is not killed. So T cell will keep on expanding and will keep on damaging the tissues. And that's how the clinical phenotype of HLH is manifested. This is the same thing that I showed in the previous picture. And if you were to look at the cytokine storm, if this was a normal response to toxin, this was SIRS, this was severe sepsis, this was MAS, this was acquired HLH, and this is the primary or familial HLH. So these, the normal immune response and genetic HLH, they are on two opposing spectrum of the immune activation. Going by the pathophysiology of HLH, you could have a predisposing immunodeficiency either by low or absent NK cell function, genetic defect of cytotoxicity, family history of HLH, prior episode of HLH, or you could have one of these markers for HLH. The findings of significant immune activation are either fever, organomegaly, high ferritin levels, elevated levels of CD25 or CD163, and abnormal immunopathology is usually manifested by either cytopenias, hemophagocytosis, hepatitis, CNS involvement, or other organ dysfunction. When you are evaluating a child, you need to ask about consanguinity. You have to look at familial disorders. You also need to look at antecedent infections, recurrent fevers, pre-existing immunological defects, any rheumatological condition, because treating that can help this patient, or if somebody was on certain immunosuppressive medicine. It is important to know about family history, because even if you lose a child with HLH, at least you are cautious about the next pregnancy, that this may have been a primary and you are at your toes. Look for rashes, look for bleeding manifestations, look for organomegaly, lymph node enlargement, look for any neurological abnormalities, these patients. How you are going to investigate, the first of all is you have to suspect an HLH, and the suspicion is usually a sick-looking child. Many times you would think of this as a sepsis or septic shock, and in fact, a lot of patients that we lose in our ICUs, which are labeled as septic shock, some of them may actually be HLH, and some can be saved if we suspected the diagnosis sooner. So you will do a CBC and DLC, you look for coagulation abnormalities, look for serum ferritin, fibrinogen. You look for serum triglyceride, and it's important to know that the triglyceride level has to be fasting triglyceride. The criteria of hypertriglyceridemia is valid if it was fasting. If you had a fat-rich meal, and most of the Indian meals are fat-rich, the high triglyceride will lose its value. Look for secondary causes. Somebody has come with fever, obviously you're going to run the cultures, you're going to look for different viruses, you look for fungus, lymphoproliferative disorders, you're going to do CT scans, and if you're suspecting parasites, leishmania, malaria, brucellosis. Many of you would have read case reports in a lot of Indian journals, malaria with hemophagocytic syndrome, leishmaniasis with hemophagocytic syndrome, tuberculosis, and a lot of other. We need to differentiate between HLH and hemophagocytosis. I'll come to that later. While we are diagnosing, we also need to do some genetic tests. They are flow-based tests, and based on that, you could direct your mutation. But nowadays, NGS-based panels are available. You could outsource this test. In Delhi, this is available at 20,000 rupees. The turnaround time for the whole panel is about 3 weeks. So you could still start the treatment and later know which mutation this patient had. Differential diagnosis would be viral infection, sepsis, a severe sepsis. Somebody could have a malignancy, a metabolic disorder. If a patient has improving without specific HLH therapy, the diagnosis of HLE is clinically ruled out. How we diagnose HLH? One, you could have a primary mutation which can cause HLH, or we need to have 5 of 8. And so far, we have been using 5 of the 8 criteria to label our children with HLH, but it is important to know that NGS panel are available in private diagnostic labs across India now. So you need to have a fever more than 38.5, a splenomegaly, cytopenias as defined here, fasting hypertriglyceridemia, or hypofibrinogenemia, hemophagocytosis either in the bone marrow or lymph node or liver or spleen, low or absent NK cell activity, high ferritin levels more than 500. Clinically, we will be suspecting HLH if ferritin levels were more than 3,000, even though the guidelines say more than 500. Generally, majority of them would have more than 3,000. If I were to run by the ferritin, this is the data of the average ferritin levels in children who were diagnosed with HLH were about 15,800. For viral infection, it was 1120. Autoimmune diseases, 1,300. Bacterial infections, close to 1,000. So for most other situations, the ferritin levels are more than 500. They are close to 1,000. And in large majority of HLH, they are close to 10,000. In fact, in pediatrics, it is said that if your ferritin levels are more than 10,000, it is literally diagnostic of HLH. I'll come to the bone marrow as one of the final. This is one of the studies from my previous hospital in Toronto. And if we looked at the patients who were diagnosed with HLH, 95 to 100% had histiocytosis morphologically. And the average number of histiocytes in per 500 nucleated cells were 6. And 12 to 14 patients had positive BMA. The median number of hemophagocytic cell was 1 per 500 nucleated cell. So you'll have a median number of histiocytes, which are 6, and median number of hemophagocytes, which are 1. It is important to know that if you're suspecting HLH and a marrow has been done and your pathologist says, I didn't find it, but there was some histiocytosis. So histiocytosis is one of the indicators. Again, go back and look at the slide that you may have missed a hemophagocytic. Sometimes it can happen that the whole slide has only single hemophagocytic cell. It is also possible that at diagnosis, hemophagocytosis was not evident, but if you were to do serial, not required, but if you were to do serial bone marrows, you will see hemophagocytosis there. Hemophagocytosis alone is not pathognomic. It is not required to diagnose HLH. You remember you need 5 out of 8. So even if the bone marrow did not show or you did not do bone marrow, you can still have the other 5 criteria, provided you are not looking at ferritin of 700, but you are looking at ferritin of 3,000 and above, you can still diagnose. Also, if you see hemophagocytosis in bone marrow, you can have malaria associated with hemophagocytosis, but that's all. You just treat malaria and somebody is going to be fine. If you are labeling someone as malaria with HLH, when all these 5 criteria, that patient is at a totally different platform in terms of survival and therapy that we are going to offer. In the large registry data, only 75% of the patients had hemophagocytosis at diagnosis. There are certain useful criteria in HLH diagnosis. The conjugated hyperbilirubinemia, transaminitis, hypoalbuminemia. So liver dysfunction is one of a very good pointers that we should think of hemophagocytic lymphocytosis. CSF pleocytosis and poor response to transfusions is one of the indicators that this child may or may not have HLH. CNS infiltration is one of the most devastating consequences of HLH. Somebody could present with seizures, altered mentation, CNS deficits, irritability, bulging fontanelle in very small babies. How do we treat this? Once you have diagnosed, HLH is a life-threatening condition. This is an emergency situation, and you need to intervene even if your diagnostic tests have not been done, even if the mutation tests are awaited, which in India may take up to 3 weeks. The broad principles of treatment in HLH are: one is suppression of hyperinflammation. So steroids are usual agent, cyclosporin is there. Elimination of activated immune systems. So again, etoposide, other T-cell antibodies, anti-infectious therapy, treat the underlying cause, very important. And at the end of it, replacement of defective immune system, which can be done by hematopoietic stem cell transplant. If you have identified EBV, treat it with rituximab. If you identified a rheumatological disorder, treat it accordingly. If you had a malignancy, treat malignancy. Sometimes you treat HLH first and then treat malignancy depending upon the clinical situation. Supportive care is of utmost importance because most of these children in ICU and most of these children can die over the next 48 to 72 hours. So supportive care is very important while the diagnostic workup or while you're suspecting it. So adequate transfusions, adequate antimicrobial coverage should be provided to these children. This is how an HLS 2004 protocol looks like. Largely, it has 3 components. One is the dexamethasone. It's a 10 milligram per meter square per day, so not a very big dose, for 2 weeks, and then half, and then half, and then half over next 8 weeks like that. Then is etoposide, which is initially twice weekly, and later it goes to once a week, and then cyclosporin, which is continuous. This is a new entry in 2004 protocol. In HLS 94, cyclosporin used to come at 3rd month. And IT therapy, intrathecal hydrocortisone and methotrexate combination is given when CNS disease is suspected. Risk factors of mortality, if somebody had hyperbilirubinemia, hyperferritinemia, and by hyperferritinemia I mean higher the ferritin, higher the chances of mortality, and CSF pleocytosis. These were the risk factors. And 2 weeks into the therapy, if your ferritin were not reducing, if your fever was not going away, if you're still anemic, very high chances of mortality. How we treat HLH, depending upon primary or secondary, the initial 8 weeks of therapy is common, whether it was a primary HLH or secondary HLH. After 8 weeks, if it was a familial disease, you continue to treat it till you do a bone marrow transplant. If it was a non-familial but persistent HLH, again, you will have to do a bone marrow transplant. If it was a non-familial and resolved, stop therapy. If it was a secondary HLH and resolved, stop at 8 weeks mark. So in secondary HLH, you're stopping therapy at 8 weeks mark. In all other HLH, you are going to transplant. If an HLH relapses, that patient needs a transplant. So bone marrow transplant is the only curative modality as of now. It's not an easy transplant to have. Indications for transplant, we have discussed the primary disorders or recurrent or a CNS involvement. Myeloablative conditions used to have very high mortality with long-term outcomes of the order of 45 to 50%. Reduced intensity conditions and smaller cohorts have shown survivals of 92%. There are certain special considerations that we need to take. The disease activity at transplant. HLH is not an easy disease to bring into remission. And if you had an active HLH at the time of transplant, your outcomes are worse. That does not deter you from, you still go ahead with transplant because without transplant, the outcome is 0%. So all efforts are done to bring the disease into remission before you could take. Even if you were to take active HLH into transplant, still we have 50% survival rates. Now, HLH can recur. This is totally odd for a transplanter. HLH can recur if somebody had a primary graft failure. HLH could recover if there was a graft rejection. HLH could recover even if there was a solid engraftment. So you could have a 95% chimerism, but in early phases, HLH can recur. It is also not uncommon for HLH patients to have a flare. So you have transplanted and you're 2 or 3 weeks into the transplant and suddenly you see HLH. This is generally not an HLH recurrence, but it is an HLH flare, it is known. The most common therapy at this point of time, most of us are used to use hydrocort or other things for these inflammatory situations, but dexamethasone is the steroid that needs to be used. You give 3 to 5 days of dexamethasone and generally this comes into control rapidly. So an HLH manifestation 2 to 3 or 4 weeks post-transplant is most likely an HLH flare and not necessarily an HLH recurrence. CNS involvement, you need a close follow-up. Intrathecal therapies is required even in post-transplant period till day 60, day 100. As the donor chimerism is stronger and stronger, the CNS disease and phenotype abates. Now, there are donor issues because one mass sibling donor is limited. Secondly, the donor may also be harboring the same mutation. And age of onset could be totally different between the two members of the family. One may present at 2 years of age and one may present at 15 years of age. There have been cases reported where from the sibling transplant was done and later sibling manifested HLH and the transplanted sibling also showed a sign of HLH. Now, if you have both parents which were low on natural killer cell functions, but the sibling has normal natural killer cell function, you can safely assume that this donor is safe, does not have any of those mutations, and you can safely use him as donor. The incidence of mixed chimerism, if you were using RIC, which is the norm these days, is generally similar if it was a matched sibling donor or matched unrelated. Now, since there can be an urgency for transplant in HLH if the disease was to fail, it is also important to know that once you are looking at a primary HLH, also initiate your donor search immediately if the sibling is not matching. The long-term data for transplants is of the order of 66% cure rates. And there was a tendency, not necessarily significant difference, in patients who had non-active disease at the time of transplant. Familial disease had 5-year survivals of 50%, and none of them survived without transplant. So you need to realize that primary HLH, if you do not transplant, we have a 0% long-term outcome. So even if the disease comes under remission, you do not transplant, this is going to relapse, the patient is going to die. Non-familial HLH, if it relapses, is close to 100% mortality if you do not transplant. So primary HLH transplant, HLH recurrence, look for a donor and transplant. We have done few cases of HLH in Toronto transplant, and I can assure it's not an easy disease to transplant. You can look at HLH worse than a class 3 thalassemia is coming to a transplant unit. Already a lot of subclinical organ damage is there. This is our data of 18 patients where we had few VODs, we had a lot of infections, bacterial, viral, fungal. We had renal impairments, children needing dialysis, a lot of pulmonary complications, including BOOP, including diffuse alveolar hemorrhages, because some of the children were also taken with the active disease as a transplant, and then other drug-induced hypertension, others. So HLH disease, even while the child is in transplant unit, is a difficult transplant to manage, but once you are day 25, day 30 of the transplant, probably you are out of woods. For our data, the median follow-up was almost 6 years, and we had 61% children who were doing well with it. So this is a treatment algorithm for HLH. If you're suspecting it, refer to a hematologist. We are hematologists. It is our responsibility to teach our pediatrician, medicine colleagues, and intensivists that such a thing exists, and urgency is the key to survival. You wait 48 hours, and probably the immunological cascade is already out of control. Child is clinically stable. Look for a triggering condition. If you have known cause, treat that. Response is well, doesn't look for other things. If an acutely ill, clinically deteriorating, use HLH-specific therapy. And dexamethasone is the steroid to be used, and dexamethasone can very rapidly break down at least the inflammation component of the HLH. And at the end of it, if it's not falling into line, you need to offer a hematopoietic stem cell transplant. So we need to realize that hemophagocytosis is a finding, and HLH is a clinical entity that needs an urgent treatment, and most likelihood a need for bone marrow transplant. You need to have a high index of suspicion unless you think you're not going to diagnose it. The easiest thing for you to do is order serum ferritin, fibrinogen level, and fasting triglycerides. You can have results within 4 to 5 hours in your lab, and you will have at least 3 criteria out of 8 that will tell you to look for more, or if these criteria are not there, your HLH is literally ruled out. So this is the fastest thing. Within the 4 hours, your lab can help you move towards HLH diagnosis or not. Also, if you are doing bone marrow is not an urgency to do. You can diagnose HLH without that. But if you are doing a bone marrow, please tell your pathologist that I am suspecting HLH, and please look for hemophagocytosis, because average number of hemophagocytic cell is 1 per 500. And HSCT is the only curative option for patients with primary HLH or secondary HLH who fail to respond to initial therapy. Thank you.
Questions are open for discussion.
I'm Dr. Fauzi. I'm from CMC Vellore. The children with HLH and CNS involvement, what would be your recommendation for CNS-directed therapy post-transplant? When will you start? How long will you give? And what?
So the idea is to continue with intrathecal. What we followed in Toronto was starting day 30, we will still give them intrathecal therapy, and we'll look for CSF. We'll look for CSF pleocytosis. That is a common finding in CNS HLH. And also if there was any neurological sign. Most of the times when a child is being taken to transplant, their systemic disease is in control, but the CNS disease might still be persistent. It's largely the pleocytosis that we are looking at. Once we see that the 2 subsequent CSF are negative, we will be stopping it. We haven't required to give it beyond day 70, day 75.
How frequently and which all drugs? Do you give hydrocot?
We are adding hydrocortisone now. Yeah.
Hydrocot and methotrexate.
Methotrexate and hydrocortisone.
How frequently? How frequently?
We were using it once a week initially.
Till the CSF clears?
Till 2 successive CSF are clear.
Suppose the CSF is clear and only the MRI is showing features?
So it will depend whether that MRI finding has been persistent for a long time or not, or whether my MRI finding was on a declining trend. If I saw that my MRI was consistently removed, I will still take it as the disease activity, the CNS disease activity, because it is known to happen. Then probably we will continue with this thing. Generally, it will correlate with your CSF pleocytosis.
No, suppose CSF is clear pre-transplant, and the MRI is showing finding which is not worsening, and post-transplant the MRI finding is remaining static. So do you need to give a prophylactic IT in the post-transplant period?
No, probably not. At that point, if it has been a stable finding, even pre-transplant, probably I'm not gaining anything by offering more post-IT therapy, post-transplant.
Okay, thank you.
And it is also important to realize that we also need to look at donor chimerism. Generally, the CNS activity goes down rapidly as the donor engraftment increases. So if day 60 your donor engraftment is solid and my CSF is clear, probably I don't need to offer that.
Thank you.
SL soluble interleukin 2 receptor, Rahul. So if the cutoff is 2400, so it has been seen in multiple studies now that if it is more than 2400, the mortality is pretty high, and if it is less than 2400, probably you are in a safer zone. So.
That was a precise and excellent presentation. Thank you. And my doubt is regarding secondary HLH. So we had two cases, one following dengue fever, and the other one, the child had chickenpox lesions. The child was deteriorating very early, and the hepatic functions were much deranged. So in such cases, can we intervene early with other agents? We have started already on methylprednisolone, but the child was not responding. So secondary agents, how early?
Once I am giving a label of HLH, once I have given a diagnosis of whatever primary and HLH, and generally these patients are sick, most of these times these children are in ICU, I am treating them. And I'm treating them now, so if you go by HLH 94, it was only etoposide and dexamethasone. HLH 2004 brings cyclosporin on board. So you could do well by omitting cyclosporin in secondary cases, just go with etoposide and dexamethasone and treat them. Once we have committed to a diagnosis of HLH, even if it was secondary, so again, it is very important to treat primary because in most situations, treatment of primary is going to take care. But if we think that the immune activation is already beyond control, my ferritin levels are very high, then we commit to at least 8 weeks of therapy.
My question to you is regarding the mutation status. You mentioned that you are screening all the donors for the mutation. So when you are screening all the donors, what if the primary does not have any mutation? In that case, do you prefer unrelated donor? Or if there's a single mutation, do you take that donor or still go for unrelated in cases where you don't know whether it's autosomal dominant or recessive?
Yeah, so there are donor issues around HLH transplants. What we would be doing is, if my donor was, so I'm telling you the clinical protocol that we would follow. HLH is a rare disease to transplant. If my donor was otherwise healthy, and if my NK cells were okay, one is nowadays, since we have NGS available, so we are going to run that mutation. If the mutation is there, that patient is out. But your question is valid that many times patient may not harbor any mutation, then it's very difficult to look for that. We'll largely go by the NK cell numbers and functions of both parents and sibling. If there's a mismatch, if we see that parents were lower and sibling was okay, it's easier or safer to take sibling. This may happen not that commonly. So you may have that the siblings numbers are also borderline, or the parents numbers were also borderline. So sometimes it may be difficult to them. Looking for unrelated donor, it takes a lot of time. So there's a practical consideration. It takes at least 2 months for us to do an unrelated donor transplant, sometimes 6 weeks if we're very quick and a donor was quickly available. But it takes at least 2 months for us to mobilize that donor, and if the donor is also ready. Many times we would actually end up using the sibling donor for transplant, but we would look for these things, at least the NK cells and the mutations.
So just one more thing, like once you have transplanted and you get the hemophagocytosis in the transplant, like in the...
If you don't mind, can we wind up the sessions because
Yes.
post-session thoughts are later. Post-session we can discuss.
Sure.
Thank you very much.
Yeah.
Thank you very much, Dr. Nathani, for
Thank
the
you so
lucid,
much.
excellent presentations.
We thank the judges and the speakers.
And we thank all the speakers for really sticking to time and all the audience for making it so interactive. Thank you.
Now we'll break for coffee. It will be served in the exhibition area. The selected scientific posters are displayed at the exhibition area. We request all the delegates to visit the poster area. And authors of the displayed posters are requested to be near the posters, as the judges will start the poster evaluation shortly. Posters.
We are on time, and it's a very interesting topic which has been kept for today's discussion. That is the sequencing, new generation sequencing is the first one. And as you know, there are a lot of progress which has happened in the DNA methodology, RNA methodology, and many of these. And this is one of the things which is for what we have progressed very well in the field of hematology. It helps us in diagnosis and also in the management. And with this word, I will request Dr. Nikhil Patkar to come to the stage and be the first batsman. Nikhil Patkar,
Hello.
please. Just to introduce Nikhil. Nikhil is the Associate Professor of Hematopathology at Tata Memorial Center, Mumbai. He received his MD Pathology from the Armed Forces Medical College and specialty training at Tata Memorial Center and Christian Medical College. He subsequently trained in genomics at the Department of Laboratory Medicine and Genome Sciences at the University of Washington. His research interests include development of novel next-generation sequencing methods for clinical application and the development of ultra-deep sequencing assays to detect minimal residual disease in hematological malignancies. So, Nikhil, I think you should
Thank you,
start
Chairperson.
and be on time so that we finish our time. Thank you. Start, please.
Thank you, Chairpersons. I'd like to thank the organizers for inviting me to this event. I'm going to talk on next generation sequencing in hematological malignancies. In the last decade, we've had several discovery studies. These told us a lot about the genome, including the cancer genome. What we subsequently understood was that between groups of patients or between groups of similar disorders, you have genes that are mutated recurrently. And this gave rise to the concept of pathway-based medicine, where we found that certain cancers have recurrently mutated pathways. This gave rise to the concept that instead of sequencing the entire genome or the coding regions, we just focus on these sets of genes, and that gave rise to targeted gene panels. Targeted gene panels are now a part of routine laboratory medicine. So depending upon what you practice, you can have lymphoma panels, you have inherited bleeding disorder panels, solid tumor panels, panels for inherited bone marrow failure syndromes, you can have organ-specific cancer panels, immunodeficiency panels, and so on. Unfortunately, in India, things are a little bit different into introducing next-generation sequencing. I work in a government center. Patients are poor. We are predominantly, patients pay out of their own pocket. The costs are remarkably high. So we wanted to introduce these next-generation sequencing-based tests. But we found that when you start using commercial panels, the cost of testing was really high, where every patient had to pay almost 15 or 20,000 rupees out of his pocket. So 20,000 rupees is not something that our patients can afford. So we decided to, when everybody tells that the biggest concern in NGS is bioinformatics, it's actually an economical problem. The major concern for implementation of this kind of testing is the high costs associated with tests. Most of these tests are driven by venture capitalists. They are geared to American markets and paying $600, where the costing of a test is $300 or $400, and what you charge the patient is $1,000. This is the norm for an insurance-driven market. It doesn't work for India, and for that reason, NGS-based testing really didn't take off in the country. So what we did in our laboratory is, when everybody focuses on informatics, we worked on the polar opposite end. We work on the wet lab aspects of next generation sequencing. So I'm going to share some of these assays with you, share the concept, and say what we want to do. So when we wanted to start developing this test, we developed an assay called MyeloSeq1. Instead of just testing for DNA and looking at mutations in DNA, we do integrated genomic profiling, where we develop testing, where we look at gene panels based on these mutations, and we do RNA sequencing to detect unknown fusions. So we have one panel that can detect that can look at the pathogenesis of all of these conditions, and depending upon what is the clinical indication that is there, we introduce the RNA sequencing for that patient. Once we identify these mutations, we look at MRD. Look at MRD monitoring based on deep sequencing, which I'll be showing you now. If a patient has ALL, we've made a panel for detecting unknown fusions, so you can identify pH like ALL and identify new therapeutic targets. For CLL, we have a panel-based DNA sequencing. For TK tyrosine kinase mutation testing, it's routine now that we do NGS based on error-corrected sequencing. The best part of our testing is that tests are affordable. For our C category patients, in-house patients, we offer this at a test of approximately $75 US, which makes sure that patients can avail these services, decisions can be made, and so on. Let's discuss this in the form of cases. So this is a 51-year-old male who comes to us with 80% blasts in the bone marrow. They're MPO positive. The FISH is non-contributory. A normal karyotype is revealed by conventional karyotyping. Immunophenotyping shows that you have a 45-dim population of blasts. These blasts show subset CD34 with heterogeneous expression of 117. They are DR positive. They show bright expression of 33, and they show NPO positivity. This is an AML. So we did next generation sequencing for this test assay, and we found that this patient harbors a type A NPM1 mutation. It's in 4 base pair insertion, one of the commonest NPM mutations that you see. So this is the NGS result. We found an NPM1 mutation. We also found a WT1 frameshift mutation and a FLT3 TKD, as well as 2 variants of unknown significance. But what do these results mean for the patient? So the new ELN risk stratification says that if you have a patient with a FLT3, without a FLT3 internal tandem duplication and an NPM1 mutation, he actually falls in a favorable risk category. And there is still no evidence in these guidelines, either the NCCN or the ELN, which says what happens if you have a WT1 mutation with an NPM1 mutation. Logically, based on what data has been shown in retrospective trials, if patients have WT1 mutation, they will do worse than this. But for now, for the book's sake, this patient gets diagnosed as AML with mutated NPM1. It's a favorable risk category. Importantly, if patients have TKD, if they have FLT3 mutations, there are some interesting therapeutic targets that are now available. If there is a FLT3 mutation, either an internal tandem duplication or a tyrosine kinase domain mutation, they result in activation of RAF and ERK pathways or mTOR pathways, all of which result in activation of transcription factors, resulting in differentiation, proliferation, apoptosis. Importantly, these patients can be treated with midostaurin, leseratinib, sorafenib, sunitinib, and these drugs are available. Just for information, midostaurin costs 8.5 lakh rupees per cycle. It is going to be available soon in India. There is data which was published in the New England Journal of Medicine, which shows that there is a benefit of 10 or 15% when you treat these patients who are FLT3 mutated with midostaurin as opposed to a placebo. This data shows that if you have a TKD mutation, these patients are even more likely to benefit from midostaurin. What else do the results mean for the patient? Well, this patient has an NPM1 mutation, all right? And data from the late Dr. Grimwade's lab showed that if patients are NPM1 MRD positive, these patients, means NPM1, when you track this mutation into the course of treatment, these patients do much worse if the mutation is detected as opposed to patients who are MRD negative. And this data convincingly showed that NPM1 is a reliable marker of AML status, and molecular relapse reliably predicted disease progression. But there were some drawbacks to this study. Number one, it's an RNA-based test. You don't want to necessarily work with RNA, especially since in our country, shipping of samples between institutes, between states is not very great. So you often receive samples that are degraded. So we didn't want to work on a DNA-based technique. Second thing is, all these assays are real-time PCR based. There are more than 50 different types of NPM1 mutations. So every time you get a different mutation, you have to develop a different assay and standardize it, which is not really great in a clinical service. So we developed a novel ultra-deep next-generation sequencing-based assay for NPM1, where in a single step, we amplify the NPM1 hotspot, or the 12, or the exon 12, where most of these mutations lie, and subject it to next-generation sequencing. We did a limit of detection testing, and we could show that we can detect this mutation at an unparalleled sensitivity of 1 in 100,000. These are duplicates of what you can see here, and you can detect this at a very, very low level, which approaches that of droplet digital PCR. So the next obvious question to ask was, do you think that an ultra-deep, very sensitive NGS-based assay will matter clinically? So we analyzed a cohort of almost 87 patients that we had accrued over the last 6 years with this assay, and we could monitor 12 different NPM1 mutations at a very high sensitivity of 0.01%. So nearly 137 samples were there, and we compared this with real-time PCR as well as flow cytometric MRD. What we found was a stunning correlation between real-time PCR and NGS MRD, but NGS MRD is more sensitive, as we could detect 10 patients here that are negative by real-time PCR, as opposed to 3 patients who are negative for NGS MRD but RQ positive. Also, there is very limited, very low bias at MRD levels when you compare these two techniques. But clinically, NGS MRD was found to be highly predictive of outcome. As you can see here, it really predicts overall survival as well as relapse-free survival. as compared to flow cytometry, there is wider separation of these curves. Also, in patients who are flow MRD negative, you can still dichotomize them by NGS assay, and you can show that these patients are actually can be split by NGS MRD status. We also showed that presence of DNMT3A mutations predict an inferior outcome in NPM1 mutated AML. With that, let's move on to the next case. You have a 6-year-old who's found to have ecchymotic patches all over body, raised WBC counts, a marrow aspiration was done, which showed the presence of 80% blasts. These are MPO negative. Flow cytometry was done, which showed a bright CD10 positive population, dimly positive for CD19, and aberrantly expresses CD33, but also shows expression of B-cell markers at a cytoplasmic level. The diagnosis is that of a precursor B lineage ALL. Cytogenetics was non-contributory. Unfortunately, this patient didn't have an approach like planned. This patient was persistently MRD positive. The first MRD at end of induction was 33%, which is very high. And subsequently it was more than 1%, and the fourth MRD is 0.9%. When patients have such a high MRD level, they are inevitably going to relapse. The reason that we know this now, that childhood ALL is actually not a homogeneous disease. Nearly 30% of patients who have an intermediate prognosis, no known genetic abnormality, and they're called as B-other ALL. There are also 11% of patients with ALL who have poor risk genetic features and are at very high risk of relapse. The current practices in Hem-Onc in our country or in our hospital is to send sample for morphology, immunophenotyping, cytogenetics for a panel of these fusions, and you'll do conventional karyotyping. But there are some problems with this workflow. What do you do about abnormalities, rest of the abnormalities by FISH? How many probes will you add? This will soon add up to the expense. Even if you get a particular fusion, what is the exact fusion sequence? How do we monitor disease when levels go down at less than 5%? What happens when there are metaphase failures? To answer these questions, what we did is we developed an assay, which is an error-corrected sequencing assay, where we basically take RNA, convert it to cDNA, first strand, then second strand, then subject this to fragmentation, end repair, A-tailing. To this, we anneal a hemi-functioning unique Y adapter. So this is a unique molecular barcode where you have unlimited number of combinations of these 8 base pairs, and these anneal to both the strands of DNA. Following this, you amplify this with a gene-specific primer from one end. So for example, if you want to amplify ABL1, it will amplify not only ABL1, but any fusion partner that is implicated in this translocation. So in this way, you can, when a library is constructed, you can sequence any unknown fusion partner. So in this patient, we did this. So we have an assay like this that detects 52 partners of translocations associated in ALL. It's a panel which can detect potentially thousands of fusions. So in this patient, we identified this fusion called as TCF3-HLF fusion. The importance of this is a very rare subset of ALL, less than 1%, but it's extremely poor. In fact, the literature states that every known patient with this disease has died, in spite of the fact that they received transplant. So this is important to know. We explained to the clinicians who told us, who counseled the patients, the parents of the child appropriately, that this is what is happening with your child, and you know, you have closure even if you can't do anything about it. To make matters worse, there's a new entity in the WHO called as BCR-ABL1 like BCP-ALL. The WHO in all its glory says that this is disease defined by gene expression profile similar to that of Ph-positive ALL. It lacks the BCR-ABL fusion. It says that the incidence increases 8. It's important to diagnose this because they have a poorer outcome, more induction failures. But then it actually goes on to say that diagnosis is complicated, requires complex laboratory analysis, and tells you nothing about how to make this diagnosis, which is very strange indeed. The reason we are interested in this is because this 30% that we just spoke about is actually blown apart because of data from transcriptome sequencing in the last decade. We know that 91% of patients who have 91% of patients who have PH like ALL as a signature show presence of these gene fusions. These fusions are broadly of 3 types. You have ABL class fusions, you have JAK-STAT class fusions, and then you have other fusions that are there. So it is important to know about them because one, this has a terrible outcome except in the St. Jude's where they have got good survival, but the rest of them don't have a great survival. It's a high-risk disease. Secondly, these fusions are targetable by drugs which are readily available with your oncologist, like dasatinib, like JAK inhibitors, like sorafenib, and so on. RNA sequencing can be reliably used to diagnose PH like ALL. This is an IKZF deletion discovered by RNA sequencing, as well as a plot that shows overexpression of CRLF2. With that, let's move on to the third case. A 31-year-old male electrician comes to us with elevated WBC counts, 50% blasts, which are MPO negative. Again, immunophenotype is that of a classical precursor B lineage ALL. When we did RNA sequencing here, we found that this patient has actually a BCR-JAK2 fusion. It is rare, with only 7 patients diagnosed to have this particular fusion in literature. And these patients, including some who have myeloid malignancies or a CML-like picture, have responded to ruxolitinib with limited duration of CR. So this is something that we need to work upon. We need to figure out how we are going to treat these patients with targeted therapy. We find that RNA sequencing brings great value into the clinic. For example, we've identified multiple fusions, including this. Look at this, BCR FGFR1. There is actually a report of this person who's responded with the ponatinib. And it is important to actually look into this, because this patient responded to ponatinib, and this particular guy is persistently, again, MRD positive, and is looking at relapse. Whenever we diagnose these fusions, we sequence them, we confirm by RT-PCR, Sanger sequencing, and so on. Again, a 57-year-old with splenomegaly, absolute eosinophilia. You can see the bone marrow is packed with funny-looking dysplastic megakaryocytes, myeloid predominance, and lots of eosinophils. This patient has plenty of eosinophils. How do you work up cases with eosinophilia? So this is a problem that we see in our clinic. We have patients who have eosinophilia, and they go around without a diagnosis. So first thing that this is easier said than done, but you look at parasitic infections, allergies, pulmonary diseases, secondary other malignancies with elevated cytokines that could have given rise to eosinophilia. If that is ruled out, you show that the patient has monocytosis, then you rule out CMML. If he doesn't have that, does he have granulocytic dysplasia? You rule out atypical CML. If not, you demonstrate clonality, and you're left with a diagnosis of chronic eosinophilic leukemia. If you can't do that, if you have end organ damage, this patient is idiopathic hypereosinophilic syndrome. If not, idiopathic hypereosinophilia. So to prove that there is a clonal abnormality, or say that this is chronic eosinophilic leukemia, you need to have eosinophilia. The WHO criteria for myeloproliferative neoplasms should not be met. There should not be gene rearrangement of PDGFR A, B, FGFR1, PCM, JAK2. There should be blasts less than 20%, no fusions that are seen, and there is no clonal cytogenetic or molecular and there should be a clonal cytogenetic or molecular genetic abnormality. Blast should be less than 2% in the blood or 5% in the bone marrow. In this patient, we did a FISH for BCR-ABL1, and it showed an extra copy of ABL1. We did a RT-PCR for BCR-ABL, and understandably, this is negative. So by then, the clinician had enough. He said, okay, I'm starting the patient on hydroxyurea. We said, okay, hang on, let's send this patient for RNA sequencing. So we did RNA sequencing in this patient, and we identified an ETV6 ABL fusion, ABL1 fusion, as a result of translocation 912. Now, ETV6 ABL1 is again rare, with less than 30 cases reported in literature. But importantly, this patients are known to respond to TKI therapy, including imatinib mesylate. After 3 months, this patient's count just normalized, and he's now in complete hematological response. It's 6 months. We have got the second sample for monitoring of this patient. The last patient is an adult male who comes to us again with lymphadenopathy, elevated WBC count, 70% lymphocytes. The immunophenotyping is suggestive of a CD19-5 co-positive population which is light chain restricted, and this is a easy CLL. But CLL has, again, as a result of genome sequencing in the last decade, we have found that there are a number of genes that we need to look at. CLL is not a homogeneous entity. It's not an indolent malignancy at all. This particular patient, we found a mutation in SF3B1, a subclonal mutation in TP53, and a VUS. This is data from on a large CLL cohort, which showed that if patients have an SF3B1 mutation, like in this case, it is a driver mutation which predicts for an earlier treatment, irrespective of IGHV mutation, deletion 7p, and so on. It's associated with poor prognosis. Importantly, in CLL, there is a new wonder drug or a class of drugs called BTK inhibitors. And there is again data to show that those patients who are treated with a drug like ibrutinib will do much better than patients treated with alkylating therapy like chlorambucil. There are guidelines in place which say now that if you have a patient of CLL, you must do two things. Number one, you must send a sample for FISH, and second thing is you must send the sample for sequencing for TP53 mutations, because these patients will benefit tremendously if you treat them with ibrutinib. So these are standard guidelines in place. So is NGS the be-all, know-all for everything? You need to be cautious when you interpret NGS results. 60-year-old male, NGS myeloid panel negative for all mutations. Hemoglobin is 18, gross splenomegaly. When we test for JAK2 mutations, we test them by 4 different methods in the same patient, irrespective of NGS being done or not. You can see that this patient has a low-level JAK2 V617F allele, which is also confirmed by real-time PCR. So you need to be aware that certain mutations are going to be missed by NGS. Last slide, and I've just, I think, exceeded my time. As a clinician, what should you know about NGS? Number one, many of people are sending samples to X lab and Y lab with commercial interests. The first thing you must know when you receive samples is, number one, what is the coverage? What is the median coverage of my sample? What is the coverage per bait? And how many baits or amplicons or whatever is used have a coverage of less than 100x? Because this is really important in knowing what has worked and what has not worked. Second thing, how are FLT3 internal tandem duplications measured? Conventional ad hoc mapping algorithms will simply miss these internal tandem duplications, and you need to have second methods or orthogonal assays in place. Third, what about CALR mutations? Notoriously difficult to detect by bioinformatic callers is the type 1 CALR mutation. It will always be missed. You need to have an assay in parallel.
Time is over.
And lastly, JAK2, how are these assays validated? And the last question is, when you start doing NGS, you're going to get patients with inherited myeloid malignancies, which you will start discovering. And you need to be prepared how to console patients, how to counsel the family, and so on. With that, I would like to say thank you. We're holding a meeting at the end of February, a meeting and a workshop. We got lots of international faculty. I would request you to come and register if you're interested in genomics for hematological malignancies. Thank you. Sorry for the advertisement.
Thank you very much, Nikhil. We have time for 1 or 2 questions, if anybody would like to tell.
Great talk. I want to ask, so when you do this somatic mutation assay, when do you suspect that it's probably germline? So do you look at a VAF, is there some, so when do you know, when are you suspecting a germline rather than an acquired?
So this is a difficult question to answer, but in a patient who's young and has a mutation at a very high allele frequency, or in a set of genes where it is known that inherited myeloid malignancies. For example, when we do testing for JMML, we inevitably say that we want to sample for germline testing. When we get a RUNX1 mutation, we invariably test for it. When we get patients with biallelic CEBP alpha mutations, we want to test for these. So these things are something that you need to look at. There's no clear answer for this. Ideally, every patient should be tested, but that's impossible in a clinical scenario. More than 50%. So 50% or 40%, anything above that, you need to get to use them.
Dr. Nikhil. Here.
Yes, madam.
So let's say I have 2 questions basically, if very basic questions. So if there are multiple markers, for example, then how and whether do you choose which one is to be followed for MRD? Does it make it cost effective to screen all post whatever for MRD? Or you choose only one which is stable?
You're right, this is not an easy question to answer. So the way we are approaching it is to, one, sequence a large population of patients in our cohort, compare this with TCGA, find out what are the 90% of mutations or 90% of the genes covered by the TCGA or by our cohort, and make a panel similar to that for testing. As of now, error-corrected sequencing for AML requires a lot more sequencing power and is not ready for the clinic. For example, I have a MySeq, but the amount of sequencing that it requires, the number of reads are in excess of 15 million, which is the read capacity for a MySeq. So 20 million reads per sample is what it takes. So we require higher instrumentation and so on. So it is not possible to select one gene or the other. We have to make a panel.
So whatever markers you get at diagnosis, you test them all for MRD as of now.
Like I said, it's a selected panel which would account for
yeah.
90% of mutations in either the TCGA or so on. It's
And
not
one point you touched in your presentation, but I just wanted to ask whether it will be incorporated in our protocols, MRD, which is based on NGS, or is it?
That is no easy question to answer globally, because what do we do if a patient has a MRD positivity? We can't refer everybody for transplant. We don't have the drugs in our armamentarium. And this is a global problem beyond which anybody can answer. So
it will evolve. Yes. Thank you so much.
Thank you.
So we have our next speaker, Dr. Prashant Embare. Dr. Prashant is clinical scientist and assistant professor at Advanced Center for Treatment, Research and Education Cancer, Tata, Mumbai. His main interest is in multiple flow cytometry. Apart from the diagnostics in hematolymphoid neoplasms, he works in MRD, monitoring in acute leukemias, then diagnosis of MDS based on flow, monitoring of myeloma, and risk stratification in myeloma precursors. Dr. Prashant, please.
Thank you, ma'am. So if you look at the program, there is one talk is NGS, then the third talk is NGS. So I feel very guilty that I'm separating two NGS people by using flow talk here. Okay, so MRD is, my talk is MRD, and it's basically focused on flow cytometry. So the people who are not who are not aware of flow probably get a little bit bored. I apologize them. I try to make it simple, and if you are from pathology background, you have already seen some flow, so you'll enjoy this. Okay. For students, initial slides are for students. Why do we do MRD? If you look at the current therapy, and if you look at the improved overall rate survival rate now, almost 90%, especially in pediatric. And if you look at two types of therapies we can give to a patient, ALL patients. One is non-intensive and the intensive therapy. If we give intensive therapy, then it increases this factor, risk factor, like cost. It increases the cost of toxicity of the drugs, then which also increases the morbidity, and it makes patient prone for secondary cancer. If you do not give intensive therapy, then there are chances of relapse. So you have to balance between non-intensive and intensive. Whom to give intensive therapy, whom not to give intensive therapy. So you have to adapt risk-adapted therapy. Now, everything we talk about in lab is, first is diagnosis, and the second is risk prediction of the risk-adapted therapy. Now, because we are moving towards the personalized medicine here. So ultimate goal is to modify the treatment based on the risk of the patient. So this is about mostly for pediatric, but if you look at even applied for adult, any risk strategy is based on three things. One is the patient characteristics, like age, then leukemic cell characteristics, and response to the treatment. So if you look at patient characteristics, is the patient's age and WBC count, leukemic cell characteristics, immunophenotype of blast, genetics, underlying genetics, as Nikhil has already shown, and the response to the treatment. So this is a standard practice, date, peripheral blood count, prednisone response, as well as post-induction MRD. Now, if you look at this parameter, if these risk factors are favorable, you get good outcome, and you can spare the patient from intensive therapy. But if you're unfavorable, you can go for aggressive therapy. And that's where the role of MRD is there. I'm not going to the literature how, because we all know that MRD is very important, not only in ALL, but AML also, and now it is coming to the multiple myeloma and even the lymphomas. So about ALL, if you look at their multiple risk factors, and this is one of the main paper from 2007 from the COG, who have defined that the MRD as well as the peripheral blood date, peripheral blood is the most important prognostic factor to predict relapse-free survival or relapse of the patient. So what is MRD? For students, this is the level of disease which is you cannot identify the disease at just conventional method like microscopy. You need sensitive methods like flow cytometry or molecular methods like PCR, RT-PCR or next generation sequencing. So just to give an example of this, this is a cartoon. This is leukemic cells you have at the time of diagnosis. This slide is taken from Sumit sir. I like this slide to explain MRD. So you have lot of blasts at the time of diagnosis. You give induction therapy. And after that, the disease will receive. Only a few blasts will remain, that is MRD, and which you cannot pick up just by morphology. Even most of the times you see blasts by morphology, they are normal, they are not residual disease. So, okay, now we have to do MRD. What are the different time points? So there are different time points, which is different practices by the different protocols. If you look at the St. Jude, they practice day 19 and day 46. Then COG, of course, day 8. So not only by morphology, they do flow also day 8 and day 29. The cutoff still we use is 0.01%. And from the European group, as well as our own multi-institute trial, that is ICICL trial, pediatric ALL, we use day 33 and post-consolidation day 78 to define the MRD levels. And the cutoff of MRD, 2 positive, we call it above 0.01%. For the sake of stratification as well as for if you want to modify it. Now, detection of MRD, if you look at the level of MRD, predict the time of relapse-free survival. So if the MRD levels is high, if the disease level is high, if not in remission, is going to relapse early. As low as you got measurable disease, if it becomes low. So if you look at, if you can detect the disease up to between 0.10⁻⁵ to 10⁻⁶, you can still predict the outcome. Below that, probably, if you do not have disease, below that, probably you cure the disease. Or at least you cannot measure that low-level disease, and the patient's immune status is taking care of the blast. Now, that does level matter. So if you look at MRD, the different levels. If you look at different levels, the outcome is different. So at each level, if higher the MRD level, the poorer is the outcome, and lower. But still, if you have negative MRD, it goes straight as compared to the other MRD. So even below 0.01%, though for practical purpose we take that cutoff, but even below that, if you have MRD positivity, it can still predict the outcome. Now, it has been shown that recently that by flow cytometry you can do the high sensitivity MRD. So if you acquire like 4 to 5 million cells by flow cytometry, you can define the MRD level as good as molecular level, or at least close to the molecular level. So flow cytometry can be used. I mean, for methods there are 2 main methods. One is multicolor flow cytometry. When I say multicolor flow cytometry, I mean more than 6 color at least. And molecular method. So in my molecular method is the qPCR1, and the second is NGS, which Nikhil just talked about. So because I'm focusing on flow cytometry, you will see in next few slides some flow charts. This is a flow cytometer. If you have not seen this flow cytometer, this is Navios from Rickman company. But yeah. Now how it works? So if you look at flow cytometer, we stain the cells by using antibody which are attached to the fluorochrome. They come in front of the laser. Here is the laser. And those fluorochromes which are attached to the antibody, they give the fluorochrome, which we filter by using different filters, and they give different information, which we evaluate. How it is done, let's see. For example, this is a cell. There are multiple antigens, so I have shown 4 antigens. Then what we do, we put antibody against these antigens. So antigens, these are the antibodies. Fluorochromes are attached to them. The fluorochromes which have different light wavelength. Now, this cell, which have attached antibodies and fluorochrome, when it comes in front of the laser, these fluorochromes get excited, and it gives different wavelength, which we can characterize by using software. So this is just a simple way to put this. Now, when we use flow cytometry, we use 2 principles to detect minimal residual disease. One is deviation from normal antigen expression. So all cells express some antigens normally. Okay. Like same way, even the precursor, like in blast, B cell precursor, those. And then other principle is to identify abnormal antigen expression. Some antigens which are not expressed, but they are expressed only in leukemic cells. So using these two principles, we detect residual disease. And these two principles covers almost 97 to 98% of residual disease. So just to show you, in BLL, this is important because B cell precursors, they are normally there. No matter what age the patient is, you will have the normal B cell precursors in the marrow. And that is the challenge, because how to differentiate between leukemic blast and normal B cell precursor. So if you look at, there are 3 stages of hematocones. This is the first stage, which is generally 34 positive and 10 bright. CD10 is an antigen and CD34, which is a stem cell marker. Then the second stage is the second stage when it loses CD34 but still retain 10, and if there is increase in the expression of certain markers like CD20. When it moves next level, they become transitional B cell, they become mature B cell. So there are 3 stages of normal B cell precursors. And because there are these stages, you have to use different markers to identify these 3 stages, normal, and to differentiate and to find out the leukemic blast from these 3 stages. Now, how do we do that? So we use different markers. These are the different markers which shows antigen expression at different level. Different level means at the strong color indicates the higher density of antigen, the light color indicates lower density of antigen, and the white indicates absence. So different markers shows different intensity, and using this feature, we can identify normal as well as abnormal. How do we do that? If you get B cells, so this is CD19, this is side scatter. Side scatter means the side scatter in flow cytometry indicates the granularity. So more the granules, higher the side scatter. So these are neutrophils, granulocytes. These are lymphocytes because they don't have granules there. And then CD19 is a pan B cell marker, so we get CD19 cells. And then we put these backbone markers, which are used to differentiate between normal antigen and normal hematocones or normal B cell precursors and leukemic cells. So how do we do that? So we put these markers and then we look into them by different combinations. So if you look at, this is normal hematocones, so CD34, this is positive in stage 1 hematocone, and they lost in stage 2 and mature B cells. 10 also shows some variation in the maturation pattern. You can see if we combine 10 and 45, it shows a very typical pattern of maturation. If we combine 20 and 45, still shows a very typical maturation pattern. And this maturation pattern genetically tightly regulated, so it doesn't change from person to person. No matter wherever you go in the world, if your reagents are constant, instrument settings are proper, you will see the same pattern throughout the person. Similarly, there are different markers and abnormal antigen expression, like CD123 normally is not expressed, CD58 normally expressed at particular level. So we use to identify, we use the cutoff. So if we see cells here, that means they are abnormal. If we see cells here, which means 58 bright or 38 reduced, that is abnormal. And so there are different new markers which we recently published, and they have shown typical pattern of antigen expression, which we use for the. So using this marker, either we use 6 color by using 3 tubes, or we can use 8 color using 2 tubes. So 8 color means there are 8 antibodies, 1, 2, 3, 4 like that, and 2 tubes, so there are 2 tubes, and 1 tube for calculation. So then we define, using these markers, we define the template. So if this is maturation pattern, what we do is put a gate around them like this. So all cells, all normal cells should fall into these gates. So anything which will fall outside will be considered in a tight cluster. If they are falling outside with multiple markers, they will be taken as a residual disease, irrespective of what was at the time of diagnosis. So this is a template we use to define, this is a simple template to show you. This is how the flow cytometry template is there, using different combination, and we then find out whether there is cluster which is constantly falling outside these gates, and then we can diagnose this as MRD. Just show an example. This is a case of BLL case. You can see the red population indicates abnormal cells. So you can see the 44 is going outside the gate. Here 45, this population is going outside the gate. Same way, here the population is going for 73, for 86. So these populations are going outside. But this is diagnostic case, so these are all blasts. But when you look at MRD level, you can see here, these populations which were outside, they are not, because the blasts which are remaining, they are coming from this part. The blasts are coming from this part. So these markers are not helpful. But when we look at 44, you can see the still blasts are outside, right? So these are abnormal. The same blast, not only one marker, but when we look at 73, as well as this other marker, 86, they are all outside, which indicates that these are abnormal blasts. We're using multiple markers, which give us confidence whether these are not normal, and we can identify the residual disease. The similar other example to see that is, you can see again the blasts, they are 24 negative here, you can see 86, they are 73 positive, and even the residual disease the same way, 73 positive and 86 positive. So this is a TMH, we do 10 color single tube, 2 tubes assay here, and last 2 years we are doing high acquisition events. I mean, we are acquiring high number of events, like more than 4 million of events, to gain the lowest level of sensitivity which we can achieve by flow cytometry. And to show an example, this was a case where we acquired 4.6 million cells, and you can see we found out the 22 abnormal cells, which comes to be like 0.0005%, which is very close to 1 in 1 million cells. So this is high sensitivity MRD, but high sensitivity MRD need a good infrastructure. You need a dedicated instrument. You also need heavy workstation for the analysis, that is computation. Just to show you a data of some cases, so we evaluated like from February 2016 to December 2017, 596 cases. And this is the level of, these are the MRD positive. So 54% cases were MRD negative. And if you look at above 0.01%, which is practical cutoff, we have 33% positivity of MRD. But still there are 14% or 12% of cases which were below that we could pick up. So you can see even the less than 0.001, there were 3% cases. And this data matches with what published by the European. You can see here, they are also 33% residual disease above this, and they have 34%, so quite matching data. So that was about BMRD, because this talk is about MRD, so I have to cover a little bit TMRD. I'm going to do that. TMRD, the TMRD is little different, because we don't have immature T cells in the bone marrow. What you have, only mature T cells or blasts, or NK cells. So how to differentiate? Because it looks simple. There are no immature T cells, so whatever immature T cells they have should be abnormal. But that's not that simple. So if you look at these are the different papers which has gone from TMRD developed from 3-4 color to 6-7 color and 10 color. So if you look at this, almost 7 to 8 color. And if you look at some laboratories like from Brainfood Lab in Seattle, they are doing 9 to 10 color MRD. So in this, when you look at the different markers, this is more little bit lab oriented for clinician. Maybe how do we do that? So when we select the antibody, we have to have one of the markers which will identify all T cells. So because almost 99.99% of TLL express CD7, so we use CD7 as an anchor marker so that we can identify all cells which possibly can be leukemia. Then we have to differentiate from the mature T cell, because CD7 is also expressed in mature T cell. So we use surface CD3 because most of the leukemia, 90 to 95% ALL are surface CD3 negative. So we use CD3 to identify or to isolate mature T cell. Then we use markers of immaturity like CD1A, CD34, CD99, and TDT. Oh, but the CD7 positive cells not only include mature T cell, it also has NK cells, it has also small percentage of myeloid blasts, which are regenerating myeloid blasts also express CD7. Then dendritic cells also express CD7. So these are the different cells which can express CD7. So you have to identify them, separate out, so that you can find out the leukemic cells. So for NK cell, for mature T cells, we add surface CD3, and for NK cell, we add NK cell specific mark like 1650, 694. We can also add additional marker like CD5 and 2, and CD4 and 8, because normal T cells has two subsets, CD4 positive, CD8 positive subsets. And when your leukemic cells are generally added into one subset, either they mostly 4-8 double negative, few of them CD4-8 double positive, and very few of them either CD4 or 8 positive. So, but if you have TDT 34 and 99, then why do you need so many markers? That is the question, right? But if you look at there has been papers published by this same COG group. They have shown that the TDT and 99 over the period, when you start treatment, they lose. They are not positive in all blasts. So you cannot identify all blasts by using this immature marker. To add to that problem, the TDT is very complicated dye to stain because some of up to 30 to 40% cases may be TDT negative at the time of diagnosis. Not because it's not there, because it's a technical issue, because antibody has to go to the nucleus level. So that is why. So that's the problem with that. So we developed a panel in 2014 where we did not include 99 TDT because of its problem. So we included, this is a 10-color panel where we put all these markers which can be useful for MRD detection, including NK cell marker, surface CD3, cytoplasmic CD3, 34, and 5, 7, 4, 8. And then we developed a template in such a way, so if you started with getting, again, side scatter means, high side scatter means granulocytes, and this is CD7. So we start with gating CD7. So we first put a gate, isolate all CD7 positive cells. Then we remove some dead cells from using forward scatter and side scatter. So forward scatter means size. So more the size, more will be forward scatter, lesser the size. So debris is generally small size, we can isolate from them.
Dr. Prashant, the time is over.
Sure. So, okay, so I'll skip the strategy, how do we do that. And so we isolate surface CD7 positive cells and exclude mature T cells. Then we exclude NK cells. Then we look at different markers, and then we can identify whether these cells are, so we can identify different subsets. And then we using CD5 and 38, we identify different subsets. And then generally, when you have these cells, we can have activated T cells, we can have MRD, we can have junk, we can have NK cells which are 1656, negative, like immature NK cells. And then using 4 and 8, we can separate them. So we can identify the disease. Okay, I'll skip these cases because of the time. And I'll show you a few slides of TMH data. So we have done a study of 160 cases. Though this data is a little crude, we have to do some cleaning, but just for this presentation, I made this. There are some cases of ATP ALL, and the majority of cases were non-ATP ALL. And then we look at the MRD level. So this were, almost, if you look at 53% cases were MRD positive in post-induction, 53.4, which matches with the data published. And out of them, in 86 cases only we have post-consolidation, so the 34% cases were MRD positive at that time. And these are the levels of, so we could even detect below 0.01% in 5% of cases. And then this is the outcome data. This is not clean, but still you can see the post-induction MRD, we could predict the outcome as compared to the negative outcome. You can see there's a wide separation between positive and negative cases. So when we look at only post-consolidation, there was some trend, but not that significant. When we combine post-induction and post-consolidation, it also shows separation, but post-induction was best. And we look at the levels, but levels were not. And this is comparable with what COG has presented in ASH few years back. So just a couple of minutes. So any testing, as other testing laboratory, need EQUAS. EQUAS means how good you are doing, whether your laboratory is doing properly or not. So last 2-3 years, we are participating in a EQUAS run by the UK and EQUAS. And after successful participation, we started our EQUAS. This is dry EQUAS, where we share the files with laboratories, and they have to analyze and give their results. And we started this year, so we completed almost 3 cycles this year. And these are the different parameters which we studied from the different. So look at how the other labs are gating, whether there is a consensus between different laboratories in gating and isolating and analyzing the MRD. You can see this is how addict. So if you look at, there are 17 labs. Most of the labs having consensus, except these 2 labs showing very low level, and these 2-3 labs very high level. Otherwise, they are showing very good consensus, indicating that many labs are doing good. So just to summarize, now MRD is the standard of care, at least in pediatric patients. They can identify MRD negative status, identify subset of errors with excellent outcome. Flow cytometry is widely used and is highly sensitive. But of course, we need adequate training. And for clinicians, it is very important when you want to send the sample for MRD, we need first pool. Means instead of making smears, because post-induction smears are useless. So instead of that, if we collect the sample and send, so first pool, first half ml sample is important, because if you collect later on, then it will be peripheral blood dilute and you may have false negative results. And of course, we need knowledge of normal patterns, therapy-related immunomodulation, and participation with ICVAS is important to check your quality and to improve your quality. Thank you very much.
Thank you, Dr. Prashant. Very informative indeed. The questions we cannot take for the want of time as of now.
Thank you
they
very
are
much.
encouraged to interact with you during dinner time.
Sure, thanks.
Thank you.
Yeah.
Now we invite Dr. Reena Das. She is very well known to all of you, but however, I will have to introduce her formally. She is Professor of Hematology in PGI Chandigarh, and her area of interest is in hemolytic anemia, thalassemia, and including the prenatal diagnosis in thalassemia. Current interest she has in NGS, in resolving difficult anemia. So it's a very new topic. I think Reena, you will all hear Reena how she has done that. Reena Das, please.
Thank you very much. My job for the next 20 minutes is to talk about NGS-based diagnosis and non-malignant disorders. And now Prashant will know why this talk is separate, because I am an outlier in this particular session. Anyway, I'm going to talk about hereditary anemias, and the diagnosis requires a stepwise diagnostic algorithm to be followed. There are two specific categories: are hemolysis, where the patients usually present with jaundice, cholelithiasis, splenomegaly with or without anemia. And the reticulocyte count actually differentiates the two groups of whether there is a good reticulocyte response or whether there is an inappropriately low reticulocyte response, which is usually seen in sideroblastic anemias, congenital dyserythropoietic anemias, and red cell aplasias, where a bone marrow examination is required. Now, the intrinsic disorders of the red blood cells are many disorders, and the globin abnormalities are the ones which are the thalassemia syndromes. The red cell membrane disorders, where the commonest disorder is the hereditary spherocytosis, and there are many enzymopathies which occur in our population. So basically, I just wanted to emphasize that the reticulocyte count is extremely important, and I always prefer doing a manual reticulocyte count alongside automated reticulocyte count, because I do believe that seeing is believing, though I know the flow cytometry people will not like that. The inherited hemolytic disorders, the commonest of course is hereditary spherocytosis, but you can see that there is a list of other disorders which are not very common in our population, but they do fall into the purview of the diagnostic algorithm. Among the hereditary enzyme disorders, G6PD deficiency is the commonest X-linked disorder of enzymes which is found in our population. The other disorders of the glycolysis, pyruvate kinase deficiency is also not uncommon. Disorders like the erythrocyte nucleotide metabolism, pyrimidine 5-nucleotidase deficiency tops the list. However, if you see all these lists, you can realize that all these diseases give rise to occur from multiple genes. So basically, you know, if you have to do a DNA sequencing and a DNA diagnosis, it is very difficult to do it as a sequencing based, you know, one by one gene kind of analysis. And that's where NGS comes very much into the foray of the diagnostic algorithm. Now, in the lab, we do all these tests, the hemoglobin HPLC. If we suspect the HBH inclusions, we look for it. If there is an abnormal peak on the HPLC, then we do the alkaline pH electrophoresis, the test for unstable hemoglobin, and the molecular test as and when required, depending on the clinical scenario. For the red cell membrane disorders, we see these spherocytes on the peripheral smear. We do the incubated osmotic fragility. Coombs test is always done to exclude cases of autoimmune hemolytic anemia, We have found in the recent years that flow cytometric eosin 5-maleimide dye binding test is a very good test to find cases of hereditary spherocytosis, and the cutoff that we take as the ratio is less than 0.8. However, we need to remember the type 2 CDA will also give rise to EMA test being within the being less than 0.8. This is the graphic representation of the flow cytometric osmotic fragility test. Now, the problems come with the enzymopathies, and not uncommonly we find that reticulocytosis gives rise to a falsely low enzyme levels. So it's among all the tests we find that the enzymopathy tests become a little difficult, not only to carry out in the lab, but also to, we have found that it may be falsely normal because of the reticulocytosis. Now, there are multiple molecular techniques, and nowadays we talk about so many techniques which are there. So we should not really jump into next generation sequencing. Many of these tests are simple tests in the lab which have their own importance, and they should be done as and when required, because these are very cost-effective kind of tests. Southern blotting is not really done in most of the labs and usually done in some of the research labs. So coming to the next generation sequencing, the workflow itself is having the genetic material, so which basically means that you have to collect the cases, because when you are putting up an experiment of NGS for benign disorders, at that time we usually like to do it as many cases together, because like that works out to be cost effective for us. So the genetic material, we get it, then it's the library preparation, the library amplification. So basically library enrichment, which is the tests, which are the procedures done in the lab. Then is the actual sequencing, which is done in the instrument. And then once you get the data, the analysis of the data. So what we had performed initially was a targeted resequencing using Illumina's TrueSight1 sequencing panel, which has 4,813 clinically relevant genes, which basically covers any of the diagnostic mutations are all there in that panel. However, if it all comes negative, it could still be a mutation, because a mutation which is not there in the panel will not be picked up by this, right? So basically we have the patient enrollment. Whenever we do the DNA isolation, it should always be done by the QIAmp DNA columns. Then DNA quantitation by nanodrop and qubit is being done. For the DNA fragmentation, we use the enzymatic degradation, enzymatic digestion. Then most of the times, because we are using as many patients, we need to do barcoding of the samples so that they can be used for demultiplexing when we do the data generation. So these indexes are added on. And then at every step, actually, we have to do the quality control, which is using the Agilent 2100 bioanalyzer. Again, and finally, we always have the qubit. So whenever we are doing the sample pooling also, we need to be sure that the DNA quantity between the samples are all similar. So ultimately, we are using the MySeq sequencing system, which is there in PGI, and the MySeq reporter and the Variant Studio version 2.1 software is used. Now, when we get the data, we get it as the FASTA files, and then the quality score is being done, which is actually the FASTQ files. Finally, we get the results as the .vcf files. After that, this is what we need to do ourselves. After the annotation of the data using the Variant Studio from Illumina, then we need to manually remove all the known variants and synonymous variants which are present in the DBSNP, 1000 genome, EXAC, and the genome ad. So using all this, when we see that the causal variant is not present in the normal population, we go by this assumption that causal variant alters the protein sequence. So basically, we are selecting the non-synonymous pathogenic variants as per the score of the polyphen 2 and SIFT, which is incorporated in through the pipeline. And then we have to look for or fish out the causative mutation. So how do we do it? We use multiple bioinformatics softwares, and most of these softwares are actually freewares which are available. So we don't have to get very, like these are not very difficult. If you do it a few times, it is possible to do them. So MutePred is another software which we use very frequently. So what are the limitations of next generation sequencing? The exonic NGS or targeted NGS panels cannot assess the variants in the non-coding regions. The copy number variations cannot be identified by this method. Insertions/deletions, which are more than 5 base pairs, may not be assessed by this method. However, we have found that even 13 base pair deletions, luckily we found in a particular case. Certain regions where a disease-causing mutation exists may not be captured, and it's important for the clinicians to understand this. And hence, obviously, if it is not captured, it's not detected. Only variants in genes implicated with the associated clinical phenotypes were analyzed, and these can be reported. The phenotype and lab investigations are extremely important to characterize the panel, which needs to be tested. Now, I'll just illustrate in a few cases. This was a 17-year male from a non-consanguineous marriage, was diagnosed as a case of hereditary spherocytosis at the age of 8 years, and he was on intermittent follow-up. It so happened that the sister, who was 8 years old, also had features of chronic hemolysis. And there was intermittent jaundice, failure to thrive, along with spherocytes and reticulocytes. And the incubator osmotic fragility was positive both in the index case and the sister. The child was on regular follow-up in the pediatric hematology clinic as well as the pediatric gastroenterology clinic. Interestingly, the sister was being evaluated for Turner syndrome with streak ovaries. Now, during the present evaluation at the age of 17 years, this patient was referred to us for the flow cytometry for the EMA dye binding test. And when we did the test, we found the test to be normal. So at this time, we had a look at the peripheral smear. When we saw the smear, we found that there were no spherocytes actually, but there were multiple stomatocytes which were there. Also, there were some giant platelets. We did the reticulocyte count, and we found that some of the very immature reticulocytes are present. This is a giant platelet which is there, and this is the platelet histogram showing these giant platelets. So this is the serial hemograms at different times that we looked at, and every time the platelets were low. This was partially because these platelets were not getting counted by the counters as the size was more. The MCV was also on the higher side, possibly contributed by spherocytosis. Because of the diagnosis of thrombocytopenia, bone marrow had been done in 2011 and was said to be having megakaryocytic thrombocytopenia. So we just Googled the two things, the presence of stomatocytes and macrothrombocytopenia, and we came across this entity, which is stomatocytic hemolysis and macrothrombocytopenia, or Mediterranean stomatocytosis, also known as phytosterolemia. The clinical manifestations in the family raised the suspicion of a syndromic stomatocytosis because the sister had features of the Turner syndromes, etc. Now, the implicated genes in this condition are ABCG5 and ABCG8, which are adjacent genes on chromosome 2, and both had 13 exons with on these transcript lengths. So after doing the NGS, we found that the ABCG5 had the mutation, which was a homozygous mutation, and this was a stop gained. And because this was already reported as a germline mutation, pathogenic, associated with cytosterolemia. So this is the integrative genomics viewer, and you can see that in place of G, this is all A, and this is actually on the reverse strand, so we would read it opposite when we are reporting it. So we did the Sanger sequencing in the index case, the sister, and the asymptomatic brother. All the three of them showed it to be a homozygous mutation. We looked at the parents, and we found that both the parents were heterozygous for this mutation. So very neatly showing that this is autosomal recessive in inheritance. And in October 2002, a cholesterol absorption inhibitor drug known as ezetimibe had received US FDA approval for the use in cytosterolemia. This is a fairly cheap drug which can be given to the patients because some of the patients can actually have premature cardiac failure, etc. We had also done the haplotype analysis, and the haplotype noted in our family was distinct from the Caucasian cytosterolemia families and healthy African American cohorts. So this is a rare autosomal recessive inherited metabolic condition in which the absorption of sterols, including both cholesterol and the plant-derived phytosterols, is unselective and uncontrolled. And these patients are said to have refrain from using vegetable oils and different vegetables which have increased oils. So I think I've skipped a slide. So coming to the next case, this was a 6-month-old Bengali child who presented with progressive pallor and mild jaundice at the age of 2 months. She was born to gravida 2 mother from a non-consanguineous marriage. There was a history of severe neonatal hyperbilirubinemia, and the child had received double volume exchange transfusion. So when we looked at the smear, we found that the hemoglobin was fairly low, and there were multiple acanthocytes which were there, and some spherocytes were also present. Now, one of our students who had passed out and is now working, Dr. Anirban in TMC Kolkata, referred this case, and when the students ring up and say that they're sending samples, I get a bit nervous because many times we are not able to offer the diagnosis. So there was significant reticulocytosis, and the special investigations were all non-contributory over 1 week period. So the plan was that I would probably do the PKLR gene sequencing because that is the commonest enzymopathy which we had not tested for. However, on doing the NGS on MySeq, we found that the gene GPI was homozygous, and this was a germline pathogenic mutation. Already had a RSID, and so there was a non-spherocytic hemolytic anemia which was there. When we looked at the polyphen 2, we found that this was highly pathogenic. And again, we looked at the Sanger sequencing. It was homozygous here, and the parents were both heterozygous. So in such a situation, we can offer prenatal diagnosis or carrier screening in the family. Now, this is just the ribbon diagram to show this is the normal kind of look of the position. The arginine 346 is involved in enzyme dimerization, and this mutation leads to loss of H bonds on the neighboring chains of the dimers. You can see over here that possibly there is a loss of the dimerization activity because of this mutation. So this is how we usually would give our reports. This is following the ACMG guidelines. And so we give the test method, etc. We also need to mention about the limitations and references. So ultimately, when you do the test and you get the results in some of the cases, about 50 to 60% of the cases do get solved, but it does take a while to, compile the report and it's almost like writing a case report and doing that much of literature search. And also the time is required because we order the primers, do the Sangers, and confirm, and every time it's like a new mutation so that we are looking at. This was a 4-month-old child from Jammu, had presented with neonatal jaundice and also had received double volume exchange transfusion. The mother was hypothyroid, antenatal period was supervised, uneventful. So anyway, the child had red maculopapular rash at the age of 1.5 months, required blood transfusion, had pallor and hepatosplenomegaly. And you can see that there was severe anemia which was there. The reticulocyte count was not too high. It was on the lower side probably. And the RBC is anyway expected to be low because of the hemoglobin which is low. So because the reticulocyte count was on the lower side, on the peripheral smear we found occasional nucleated RBCs. So we thought that probably it's some kind of a congenital dyserythropoietic anemia. The bone marrow examination showed all these erythroid cells and some degree of dyserythropoiesis, but this was not very significant. So this is the bioinformatic analysis which was done after the MySeq, 11,000 variants. Then after the synonymous variants are removed, 7,222 variants. After the normal variants, normal polymorphisms are removed, 4,194 variants. And the polyphen 2 and SIFT predicted it to be deleterious in 15 variants. And then we just looked at the candidate genes. And out of the 45 candidate genes, we looked at 16 genes, and we found it to be the PKLR to be heterozygous. So after seeing that the PKLR is involved, we did the Sanger sequencing. And in the Sanger sequencing also we neatly found that the patient had a novel mutation, and this is a mutation which was already described. So one was segregating to the mother, the other was segregating to the father, and showing it to be an autosomal recessive kind of inheritance. This is a case of hereditary spherocytosis, where on the genomic DNA we found the mutation, which was initially identified by NGS. On Sanger's, we found it to be a heterozygous mutation. The ANK1 usually presents as an autosomal dominant kind of inheritance, and then we ended up doing the cDNA sequencing of the reticulocyte mRNA. And in the cDNA, we found that there were 2 bands which were seen in the patient when we compared it with the mother and the father. And when we looked at the cDNA sequencing, we found that there was an exon skipping of the complete exon of exon 4, which was 481 base pairs. So the take-home message is that for hemolytic anemia's diagnosis, it requires a stepwise diagnostic algorithm to be followed. You have to be patient for every case actually, because sometimes it takes a few months to come to the diagnosis. The clinical manifestations are usual. The phenotypes can vary from fully compensated hemolytic anemia sometimes without even having anemia, just having reticulocytosis, and the patients are getting investigated just for jaundice. So the causal genes implicated in congenital hemolytic anemia are numerous, making a gene-by-gene approach not only time-consuming, expensive, and labor-intensive. The use of targeted resequencing can expedite the molecular diagnosis, where the cause for hemolysis remains unexplained after routine lab tests. The family studies are extremely important to prove the pathogenesis, including the unaffected family members, which we give a lot of importance because they also help us to tell that this is probably a polymorphism. So the normal DNA samples are also important apart from just the parent samples. I would like to acknowledge this work, which was done in collaboration with the pediatric hemato-oncology clinic, the adult clinical hematology unit, my PhD students, Ms. Manu and Anu, and Dr. Arindam and Professor Partha Majumdar from NIBMG did our initial hand-holding for us. And we also used the exome sequencing. We did some of them in some of the cases from NIBMG Kalyani. The DM students who always remain inquisitive to ask questions, and the excellent technical staff of the Institute. Thank you very much.
Thank you very much, Reena. It's a very nice presentation, and unfortunately, there is no time for taking up the questions. So, ladies and gentlemen, you've enjoyed this session and saw that how this progress in the molecular method and genomics has helped in the diagnosis of malignant and non-malignant hematological disorder and has helped the therapy in those cases. Thank you very much. We also thank the organizers to give us this opportunity, to myself and Anjali to be the chairperson. Thank you.
Thank you, chairpersons and speakers. With that, we come to the end of this last session. We request all the delegates to take a short break of 10 minutes while the hall is being rearranged for the inauguration ceremony, which will begin at 6:30 p.m.